All right, thanks guys for having me. This is really a good time to go first, I think, for my circadian rhythm, flying up in the UK. And as I told Eileen, I was really excited to get this invitation, but I felt like I didn't have any cutting edge biomarker work to present for you guys. So I'm very excited to be here and listen and be part of the discussions. But especially because we are working on creating more of a biomarker network in Europe, Oliver and Cahill and other folks. And so I thought I would talk about something that might just generate some discussion and ideas. And this topic, GLP-1 medications, so the new diabetes, anti-obesity drugs. I should disclaim that I don't have special expertise. I'm kind of a demographer or social scientist who has worked a lot with biological data.
And a nerdy girl.
And yes, and some science communication during COVID and nerdy girls. But it's something that I've just been approaching because I'm very curious about it. Trying to read a lot and think about what it might mean for society, population health, but also more specifically the type of questions and research that we all do. So you guys know very well kind of the status quo here in the US. We've gotten to a place where it's unusual to not be classified as either overweight or obese in your BMI category. We've also at the same time seen these dramatic slowdowns and improvements in mortality that have been attributed a lot to cardiovascular disease. Deaths of despair have gotten a lot of attention, but we know that a lot of the slowdowns in the US are due to CVD. And we can't draw a straight line necessarily from the obesity epidemic to those slowdowns in mortality, but it's certainly plausible and things that we're looking into. And then despite all of our, efforts, perhaps not best efforts, we have been unsuccessful at reversing these upward trends in obesity at the population level. And that's really true globally. There's been no country that has, reversed or even kind of halted these increases in obesity. So seemingly, we need some help. And that's kind of where these new medications might come in as potential breakthroughs. Oh, I'm sorry. There we go. So I think this is where it suddenly feels like there might be something on the horizon that completely changes the game. That could be too much hype, but I think we're seeing some really, really intriguing things. There we go. And I'll just say, some folks, such as John Murdoch at the Financial Times, even think that we may already be seeing the impact of these drugs on population obesity. I was a little more skeptical about how, the error that, within the error in NHANES that we could conclude that already. But certainly not seeing the line continue to go up could be considered a very good thing. So what's the usage? It's obviously in the news a lot, and we see all the advertisements on TV. But as of this Kaiser Family Foundation poll from 2024, May of 2024, about 12% of US adults had said they'd ever used GLP-1 drugs. And current use was around 6% for comparison. But you can see within these other categories that it's a much higher percentage of people whose doctor has told them they have diabetes or other conditions. And then I don't have it on here, but for ages 50 to 64, the overall ever taken rate was 19%, although for over 65s it was a bit lower, 9%. And so this is, I guess, not huge prevalence right now, but I think, it's safe to assume that as especially indications and approvals, get even larger, I guess we'll see with the current health secretary, who might not be as enthusiastic about these drugs. But I think we can, expect prevalence and usage to increase a lot. You guys probably know kind of more about the biology of these drugs, but very briefly, these class of drugs mimic the glucagon-like peptide hormone. which enhances insulin secretion, suppresses glucagon release, slows gastric emptying, promotes satiety. And so, it was really designed originally for blood sugar control, but we saw, as we well know, kind of dramatic weight loss that accompanied use of these drugs. And so that got a lot of people's attention. And now we're starting to see from the trials and also more observational studies a lot of potential impacts outside of even diabetes and obesity, cardiovascular disease, and we'll talk about other potential aging outcomes. And so, I'm really trying to think how these drugs might impact trends in aging and mortality at the population level. And I saw this just on LinkedIn a couple of days ago, so I thought, if the people who have, are putting down money, I guess, on these mortality trends are thinking a lot about it, then it probably means that there's, something important going on. And in fact, we are seeing, beyond diabetes, significant reductions in risks like cardiovascular disease, incident events, as well as all-cause mortality. There was a paper a few months ago, because the trials were running during COVID, that showed lower infectious disease and COVID mortality rates in the treated groups. But we've now seen papers, I think, on renal disease, heart failure, lots of other outcomes. And I think, intriguingly, a lot of these benefits, at least, are suggested to be in at least net of the weight loss itself, because we know weight loss would contribute to a lot of these improved outcomes, but there seems to be effects, direct effects above and beyond the impact of weight loss by itself. I think the other thing that I've found interesting reading these papers is that even things like adverse events, so everything that a clinical trial has to keep track of, sort of anything that happens to these folks over several years of follow-up, adverse events are actually, less common in the treatment group, which I think makes sense that it's affecting outcomes in a lot of different systems in a lot of different ways. And this was another thing that really caught my attention, as I'm sure it will for a lot of folks here who've thought about inflammation. Not surprisingly, I think we see some big reductions in biomarker risks that we're accustomed to collecting, like blood pressure and HbA1c, but there's a big effect on CRP in these trials. And again, there's, I think, in a series of papers, arguments that this is above and beyond the weight loss itself, because not everyone loses as much weight. So you can kind of see that it happens kind of even before weight loss really happens. So there does seem to be evidence of anti-inflammatory pathways that I think are really exciting. And we're also seeing, some more real-world studies, some observational studies. So this paper just recently came out looking at the veterans database. So over 200,000 people in that database who had been prescribed GLP-1s, and then the comparison was people prescribed kind of alternative, I guess, diabetes drugs. And then there were still a lot of matching things, but it was observational. And the intention was to do discovery of across 175 different outcomes of what differences might be in people who are on these drugs. And this is the kind of list of things that came out better for people who are on these drugs compared to an alternative diabetes drug. So a lower hazard to the left of one there. I guess I cut off the values, but you guys get the idea that these kind of lower risks are seen across a variety of systems and outcomes, things even like septicemia, but you'll see in the mental nervous system disorders, alcohol use, schizophrenia, suicidal ideation, all sorts of really interesting results here. And I think they did do, outcomes that they wouldn't expect to be impacted, just to check for confounding, and it seemed to be robust to that. There were some things that, popped out as higher risk, not surprisingly, I guess, but they kind of make sense, things like low blood pressure, digestive system kind of symptoms, passing out, things like that, sleep disturbances, headaches, and then I guess the musculoskeletal is interesting because there's some evidence that it's good for bones, but that's something to keep an eye on. But you can kind of see that on balance, there's like a lot more things that are very serious that seem to show a positive effect in this really large sample. And I think the effects on things like alcohol and addiction are super intriguing, I think, to a lot of us. I think we don't really know what's going on there yet of how these drugs are affecting the brain and the reward system. But this was just this week as well. And crap, yeah. I usually drink wine to reduce alcohol. It's my favorite one.
I found Jesus.
But a lot of people are excited about that. There are trials. Some of the trials right now are testing these for addiction outcomes, which I think is really, really fascinating. And just to kind of throw out some, the literature is moving really fast, and I'd say a lot of it is still very much animal or in vitro models compared to the stuff we like in populations. But there seem to be a lot of potential biological pathways through which these drugs work that are not just metabolic regulation. Some really good evidence for lowering inflammation systematically and in tissues. I think some suggestive evidence, again, more in animals, for potential effects on DNA methylation, autophagy, and then, really, for me, also exciting suggestive evidence about these drugs being neuroprotective. There's receptors in the brain, they cross the blood-brain barrier, and some really exciting, I think, suggestive evidence and also trials for Alzheimer's and dementia that are happening right now. And also, I should say, Parkinson's. So there's a lot of kind of neurodegenerative diseases that are seeing some promising signs and being investigated. And I think for folks here, there's a lot of kind of the omics work that we're starting to do in the population level that is also being then taken to kind of trace out the pathways through which these drugs might work. And this was a pretty recent one on that used trial data to look at proteomic signatures in people who were treated versus placebo. And you can look at the paper, but they did find a whole bunch of differentially effect or protein targets that were significantly affected by semaglutide in this case. Down-regulated proteins involved in key pathways, including mTOR. And so, I think there's a lot of potential also in thinking about how this trial data perhaps might be leveraged and repurposed to investigate some of these specific biological pathways. And I think I've heard Dan Belsky might be up to some of that work for the biological aging, but I think there's a real opportunity there. And, I was trying to find really, papers that were looking at this specifically with aging mechanisms. And this paper is a preprint in bioRxiv, but, kind of did all the omics in mice, trying to see if this could be considered a broad counteracting of age-related changes. And so I'll let you look that up if you're interested. But they did find significant effects even in young mice and kind of enhanced cognition and a lot of interesting things. And in this mice model, they were able to induce these changes without weight loss necessarily. They found a dose that did not induce weight loss, which I thought, again, from an aging standpoint, could be really important in humans. Obviously, kind of like the old biomarker hype curve that Coulter used to talk about, this feels like, it's really, really hyped, and every paper that comes out just almost feels too good to be true. So we obviously need time, I think, for some of this data to play out, but even things that we know we need to be mindful of. Obviously, people losing a lot of weight also lose muscle mass. I think I've seen some say that it's above and beyond what you would expect from normal weight loss, but I don't know if that's 100% established, because obviously any weight loss is always a mix of lean and fat mass. But as I was saying, a lot of older people don't need to lose weight, so can there be potential benefits, for everyone that could be leveraged without weight loss, I think is an important question. I think we need a lot more kind of long-term follow-up. But kind of as these data mount about the benefits, I would think any kind of unforeseen long-term negative impact would have to be very large to kind of counteract some of the benefits we're seeing. And you probably know right now it looks like you kind of need to stay on these drugs to maintain at least the weight loss benefits. And a lot of people don't tolerate them well, don't continue them for that reason. But I think it'll be important to know what happens then with discontinuation, especially if you've lost a lot of weight, and we know that regaining weight, before these drugs, just in other types of massive weight loss is really can leave you in a worse place metabolically than if you hadn't lost that weight. So I think that's something we need to think about. I think questions for us, the type of work that a lot of us do is to, yes, kind of think about can these GLP-1 therapies reverse aging or, our markers of biological aging in a way that we can detect. And I've kind of been thinking about, if we are seeing, huge improvements in all these biomarkers for older people, how much will that kind of set their disease risk, back to baseline as if they had never been obese or had diabetes for decades? I think that's something that's probably not going to come out of the trials or those types of very clinical investigations, but we should try to think about, how much is this going to really alter people's trajectories towards chronic disease and disability? Does it depend on the age at which you first get this intervention, et cetera? And as I said, I'm really, really excited, and I think lots of people here who work on Alzheimer's and dementia will be kind of waiting with bated breath for these trials, specifically on that outcome that I understand will be reporting out in 2025. And finally, I think also, thinking about what these medications might mean practically for our population-based studies. I was talking with Jennifer at breakfast just about, do we have to think about additional questions and measurement beyond kind of that prescription list? Because this feels a bit different than just statins or antihypertensives. It really feels like understanding what people, how long they might be taking it, did they try it? Just a lot of things that we might need to anticipate and consider. And I also think that the types of studies in the family, HRS family studies, could be really well suited to study some of these more population-based implications about whether we're changing people's trajectories and how that depends on a whole series of life course, social, and other health exposures, et cetera. But I kind of, given the current prevalence, I don't know how quickly that will go up. I am wondering, will there be just enough people in the samples to kind of analyze those types of impacts? From the social science side, I haven't said a lot about that, but we know there's going to be big issues with access, paying for these medications right now, equity, that I think we all need to discuss. I will say there's tons of these drugs in the pipeline. So I think it's reasonable to assume over some time frame that tolerability will improve and prices will come down, but that's not automatic for sure. And then kind of really zooming out, these are the things that I think I'm interested in understanding. Will these drugs really finally turn around that obesity epidemic and bring on maybe a renewal of that cardiovascular revolution where we saw really rapid improvements in mortality? And then if we do, are those really coming from reducing risk factors that we've put upon ourselves, kind of the man-made stage of the epidemiological transition? Or is there a chance that these are one of the things we've been looking for to slow down that aging process entirely? And then given all these, widespread effects, but especially I think on the behavioral addiction, all sorts of things that we didn't expect, what might the economic and social implications of widespread use of these drugs be? To me, there's kind of a million different questions that could come out of that. And this was also a very recent thread on Blue Sky by Angie Rasmussen. The whole thread is really great. She's a virologist, but she kind of tells a bit of her story. And, besides just, the physical effects of feeling like she looked better, she had all these blood labs got better, but she drank less, she had more energy, and she just kind of said how it was really important in improving her mental health in and of itself. So I think there's just a lot of interesting implications that we haven't fully thought about. So yeah, I'll leave it there for discussion. I think these are just really, exciting, potential game-changing drugs. But it is very, very early days, and I just kind of want to encourage all of us from this more population perspective to get in there early and think about these questions and anticipate data needs that we might have, new questions that we might need to ask in our surveys, et cetera. So I'll stop there. Sure. Do you want to just shout it out?
Mara,
yeah.
I was really struck by was it 1 in 8 are on it?
That was have ever taken one.
Yeah. And how accurate are those numbers? Where do they come from? How does this get tracked?
That was from a survey from Kaiser Family Foundation. So they do a lot of kind of good surveys about current health behaviors, but I guess I don't know exactly how they draw. I'm sure they're aiming to be somewhat nationally representative, but it's not just a convenient sample. So it was, some kind of survey-based poll of the nation at that time.
It's really amazing.
Oh, Jennifer, and then.
Well, as I told you this morning, I think you always do such a good job trying to keep population health scientists modern and in the moment. And I was excited that you were giving this talk. I just wanted to add something on there, which is that I've recently become aware of a phenomenon that I think our biology colleagues have known about for some time, but there's a growing group of individuals, particularly in the United States, but in other countries, who are now trying to biohack their health, aging, and longevity. So in addition to these GLP-1s, which some people will microdose, meaning they take a subclinical amount, women are doing it in menopause to reduce symptoms of menopause. But in addition to that, people are taking other peptides. I don't exactly understand where they're getting it from. People are taking MOTS-C somehow. Men are on TRT. Maybe women are taking TRT. And so, our science and the biology of aging has fully jumped out of the laboratory and into the public domain. And because you think about these things and you think about packaging and communication, I was just wondering if, have you, and this guy, Steve Johnson, is the best example with his ridiculous epigenetic aging, but have you thought about, I'm curious what your thoughts are about this complete fusion now of our world with the real world.
Yeah. I guess I feel like you guys are in the epicenter of a lot of that even here at USC. So I'd love to hear, I guess I haven't thought about it a lot because I assume that the prevalence of that type of biohacking is very small and an elite group, but it has occurred to me in the current climate that it could be a good marketing thing for the tech people who want to invest in health research that, if anything is anti-aging, it's probably more palatable right now than, just our normal concern about improving life expectancy, et cetera. So, and I wonder if that's true broadly in the sense that people are kind of bored with the way we normally talk about it. It might be a good way to communicate more broadly, if that is what's kind of speaking to people. But I worry a lot about the stuff that those people are selling online like that. It seems like a whole big racket that we wouldn't want to feed into necessarily. But if we can co-opt that somehow.
So, do you know anything about the distribution across other countries like England and Ireland? Of the use of this, since your numbers are surprisingly high to me, even for the US. So
I should get a handle on that. I think it's definitely lower in the UK because the NHS has much more constrained.
So it was 40% died, 43% died.
I have people in the family who take these drugs.
Yeah.
Who've even over Christmas ate extra cheese just to get their BMI to a level where
But
the
it's 35 and over BMI, I understood in the NHS.
Oh, if you were high enough, you can get them.
but this is the first time in England where
Okay.
people are getting drugs not through the NHS. We're so
Okay.
used to going through the NHS, but actually these are sold commercially through pharmacies, through the supermarkets. You go into the supermarket, have a sign saying "Sign up to our new thing, our new weight loss." So it's really
That's
Okay.
out of the bag.
it's
I'm gonna have to get up
heavily
on that.
promoted.
But my sense was countries where it's more of a nationalized health system have not, it hasn't gotten quite out as much, but I think
No, but it's
sorry,
private.
Lauren.
I had a couple, yeah, fascinating. Thank you for bringing this up. And so a couple questions. First, I've heard there's something with dosage that's in terms of how fast the weight loss goes. I have a friend who opted to do kind of a more minor dosage and lose weight more slowly versus a more major dose. And in part, she's hoping then when she steps off these drugs, she doesn't get a rebound. So I don't know if like there's anything there that you think is interesting. And then the other question is I've heard that some people have pretty significant side effects in terms of nausea, and they're just not able to take the drugs, kind of like naltrexone, which also has, of course, anti-alcohol addiction, similar.
Okay.
So, and it tends to be people perhaps with certain genes, I've heard, who also experience things like
Hyperemesis.
Thank you, yes, in pregnancy and things like that.
Okay.
So just curious if you
Yeah, I
heard
think-
that, population differences there.
Yeah, I glossed over a lot. There are a lot of people who discontinue. I can't remember if it's like 30 or 40% of people who try, but just where the digestive stuff is too much for them. And I think apparently we have gotten better at kind of laddering the dosage so that that's minimized. And then there's also some people who don't lose weight. Like there's a distribution. There's a paper that kind of shows the distribution of weight loss, which is really interesting. And wouldn't you feel awful if you went through that and didn't lose the weight? So I think there's tons of heterogeneity things to study, but I also think there's so many in the pipeline right now that, and kind of different mechanisms even that, and pills. And so I kind of feel like that's going to get a lot better, but I can't say that for sure. What was your first question?
Oh, just about kind of the, like, whether you read a lot about people regaining the weight after
Right,
they
and I think,
went
yeah,
off.
that's a big thing. Apparently discontinuation, I think some people, I don't know that we have really great studies on that yet, but people, tend to regain. So the downside might be if you have to stay on it forever. And will people do that? I mean, it could be like they do with their other anti-hypertension meds and things, but it's a drawback for sure.
Thanks for the invitation. I'm going to talk about some ongoing work in our lab where we are interested in studying immunity and its role in aging. So this is ongoing work, and I'm happy to take any feedback people may have on how to better analyze this data. So background, we measured various numbers of T and B cell subsets in HRS. We measured around 14 different T and B cell subsets. And we published several papers showing their effect on CMV, showing their association with socioeconomic status, age-related diseases, mortality, so on and so forth. The problem with the data, well, it's great. This is one of the few studies that actually have this data in this depth. And while we have published a lot of papers, the limitation of this data that we have is that what we have information is really on the cell distribution, the numbers of cells. We don't have any information on how the cells function or the quality of the immune response that is there. So we've been thinking about how to use existing data in HRS to expand, what we can learn about how the immune system functions. So before I go into, what it is that we've done with the HRS data, the next few slides are going to be a, immunology 101 thing so that people can orient themselves to what it is that I'm going to be talking about. So the basic function of the adaptive immune system, which has the T and B cells, is to recognize a foreign antigen. The way we do that is by having these molecules, which are called immunoglobulins. Now, if the immunoglobulins is secreted by a B cell, floats off into the plasma, we call that an antibody. If it is anchored to the surface of a B cell, we call it a B cell receptor. Now, B cell receptors are one of the most well-studied molecules, earliest and most well-studied molecules in the immune system. These receptors or antibodies have two functions. One, they need to bind to the antigen, and second, they need to destroy the antigen. So the parts of the antibody or the immunoglobulin that bind to the antigen are called these variable regions, colored in green. And then the parts of the B cell receptor or the antibodies that attract the other effector cells, mast cells, platelets, complement cascades, to come and destroy the foreign antigen. That's called the constant region. So these are the two functional pieces of an immunoglobulin. There is a variable region that binds to the antigen, the constant region that attracts all the other cells. Now, structurally, you can divide this into, for the B cell receptor, you have a heavy chain, which is this, the longer chain here. And then there are two light chains. There's a kappa chain and a lambda chain. The kappa gene rearranges before the lambda. So in general, in most people, not most people, everybody, there's twice the amount of kappa light chains as compared to lambda light chains. Now, when it comes to T cell receptors, the idea is the same. They have variable regions, they have constant regions. The same idea as the B cell receptors, but instead of the heavy chain and the light chains, you have alpha beta chains, a combination of alpha beta chains that account for 95% of all the T cell receptors in the peripheral blood. And then there are gamma delta chains that account for a small percentage. The big difference between T cell receptor and B cell receptor is that TCRs, they do not directly interact with the antigens. Instead, there's a complex, there's an antigen presenting cells that expresses these major histocompatibility complex or the MHC molecules. And so it's a combination of the MHC molecules and the antigen peptides that are recognized by these T cells. So when you want to think about all the we want to think about the diversity of these receptors. What we want to measure is the sum of all these T cell and B cell receptors in the individual. Now, how many are there? Within each individual, the total number of T cell receptors exceeds the number of people in the world. So there are 10 raised to 13 estimated to be around 10 raised to 13 receptors just for the T cells alone. So it's a highly complex system, and this is the reason we have the luxury about talking about aging, because these things exist. If they weren't there, we would die very young. So how do you measure this? We start with DNA or RNA as the starting material. RNA is preferred because, when you start with RNA, you get to the receptors that are functional and are expressed on the surface. When you start with DNA, there are a lot of non-productive recombinations that you pick up as well. Then the second step here is to perform some kind of a target enrichment, so that you select the sequences relevant to the T cell and B cell receptors, and then you perform RNA sequencing on them. There are various methods to do this. We did not perform targeted enrichment in HRS, but we did do whole transcriptome sequencing. And that is the basis for which we were thinking of using the whole transcriptome sequences that we had in HRS to get an estimate of the T cell or B cell repertoire. How do you analyze this data? There are actually, so this is common in the immunology field, but not so much used in population sciences. There are multiple software that have been developed to take RNA sequence data and convert and get an estimate of the T and B cell repertoire. Mixer is one of the most widely used software, just based on the number of green check marks you see. It has more functionality, but more important, it is very fast as compared to the other software. So that's the reason it's gained a lot of popularity. What does Mixer do? You take your raw sequence files, your FASTQ files, you align it specifically to the V regions, the C regions, and there are diversity and junction regions as well. You specifically align it to those regions, and then take them and assemble them into what's called clonotypes. So each clone has the same T cell or B cell receptor that is being expressed. So you just count the number of clones. So all these three steps is done by Mitzi. So once you do that, what it is, how are we going to evaluate these clonotypes in in population studies? So this is an example of the T and B cell repertoire that are there in the human body. Whatever method we use, be it some targeted method or this whole transcriptome, we are never going to be able to get all the 10 raised to 13 T cell receptors. We're going to get some subset of that. And we are going to get two types of information. One is we can look at the number of unique clones. So whether a person has 10 different clones or 20 different clones, we can get that. And you can also count up the total number of clones that are identified by sequencing, and you can get a total number of clones. So this is how a mixer output looks like. It comes out as a tab delimited file for each sample. It comes out as a tab delimited file, and you can see there are 6 tabs per person. So you have IgH, IgK, and IgL, which are the B cell receptor clones. And then you have TRA, which is the alpha chain, TRB, beta chain, the gamma chain for the T cell clones. What you get is clone ID. You can just add up all the clone IDs, that's the number of unique clones. Even though we are using RNA as the starting material, you get some receptors that have a stop codon in that they don't actually produce a receptor. So we remove all these non-productive clones, and then you can add up the total read count and get a total number of clones. So these are the two, so you get 6 different outputs. Each of them you can have like 2 variables that you can analyze. We've done quite a bit of work. We don't really find anything interesting with the total number of unique clones. It's the total number of clones in a person that we find some association. So after a lot of work, after many years of work, we were able to finish up, this mixer analysis, use mixer to analyze all the 3,700 RNA-seq files that we had in HRS. We processed it all to obtain 6 different, the 6 different types of clones. The 3 B cell receptor clones and 3 T cell receptor clones. Now this is the distribution. So I want to, there are a couple of interesting things here. One is I mentioned a few slides ago that there are 10 raised to 13 different TCR clones within an individual. The x-axis here goes from 0 to 135. So that tells you that we are picking up only the dominant clones. We are missing a lot of the minor clones. But there are some patterns that give us hope that this actually works. If you look at the TRAD and TRB, I mentioned that the alpha beta chains account for 95% of all the T cell clones. The alpha and beta receptors are actually much higher than the gamma receptor, the T cell receptor. So fits with what we know. And similarly, when you look at the B cell clones, I mentioned that kappa chains are typically twice the amount of lambda chains. And you see that rough proportionality seen here as well. So this tells you that even though we are picking up only the dominant clones, the relative abundance is slightly preserved. Now, when you look at the correlation between all these clones, some reasonable patterns emerge. We know that the alpha chains and beta chains always, the receptors are formed together. So there is a very high correlation between the TRB and the TRAD receptors, 0.85. Lower receptors on the other thing. Our hypothesis when we went into this project was to say that, the higher the number of T cell or B cell clones that we detect would correspond to a higher level of antigenic exposure. So that was the idea, that we could use this as a metric for total past antigenic exposure. Now, when you look at the first column, which is age, that tells you this is the first indication that my hypothesis was falling apart. Because you can see that these T and B cell clones actually had a negative correlation with age. A small negative correlation, but a negative nonetheless. Next, we looked at how these clonotypes were associated with standard demographic factors. This is race. All the T cell and B cell clones were higher in blacks as compared to the other races, and significantly. And then when you look at sex differences, the T cell clones were higher in women as compared to men, but there's no sex differences in the B cell clones. Next, when you look at, next we wanted to look at clonotypes and how they were associated with risk factors that represented ongoing antigenic exposure. So you have CMV status in the clone. When you look at CMV status, the T cell receptors were higher in the CMV positive individuals. And this is a good negative control. You don't expect the CMV status to be associated with the clonotypes. And so we did not find any differences between B cell clones and CMV status. Next, when you look at smoking status, again, what you see is only among current smokers, the diversity of these T cell clones was higher as compared to both former and never smokers. And interestingly, it is almost exactly the same in the former smokers and never smokers. So suggesting that this is likely, what we are seeing, the higher number of clones we are seeing in the current smokers probably represents kind of ongoing antigenic stimulation that we are picking up. Next, we looked at these correlations of these clonotypes with other things. We have created things like comorbidity index, which is a combination of all diabetes, hypertension, stroke, cancer, so on and so forth. So we created that index. We have created a surrogate variable for inflammation that takes into account CRP, IL-6, all the interleukins that we have measured in HRS plus CRP. So we created the surrogate variable and then BMI. And interestingly, all these T and B cell clones are for most part either not associated with these markers of inflammation and other things, or they were negatively associated. And then next, we actually looked at the correlation between all these clonotypes and the immune cell subsets that we have measured in HRS. So the left is what you see is the correlation between T cell receptors and T cell subsets. And the right is B cell subsets and B cell clones. Just by looking at the color, they're all positively, weakly but positively associated because these receptors are present in all the T cell and B cell subsets, be it a naive cell, a memory cell, or an effector cell. They all have T cell receptors. So you see a positive association with all of these. Next, before we did some substantive analyses, we looked at how reproducible these clones are. So we had in our RNA-seq data, we had 32 pairs of blinded duplicates. And so we looked at what's the correlation for these 6 clones. And in general, they are more or less reproducible. Your correlation ranges from 0.7 to like 0.9 plus. There are some clones like the TRG, the gamma chain, which has very low frequencies, where you don't have a nice correlation, but for most part, this is good. However, when you look at the reproducibility of the individual clones, if you look at the sequence of the clones, you don't get great reproducibility. And initially my thought was this is related really to the sequencing error that we have in RNA-seq. But even the more targeted methods for looking at the T cell and B cell clones don't show reproducibility much better than 30%. So suggesting, I'm not sure what it suggests. I mean, what it suggests is, we shouldn't be looking at individual clones. We should be looking at least in population studies, we should be looking at the numbers of clones. That's fairly reproducible. But we shouldn't get ahead of ourselves and start looking at individual clones. So next, finally, we did an association of these different clones with mortality. This is 4-year mortality in HRS, so mortality assessed up to 2020. The clonotypes are measured in 2016. We did some basic adjustment for age, race, sex, education. And then, importantly, Mixer does not adjust for the total number of RNA-seq reads. And so we put that into the model as well. The blue line represents the baseline model, and this is the full model. The red line is the full model. And you can see the T cell receptors, beta and alpha, were negatively associated. These results are presented as one standard deviation change in these clones. You see that they are negatively associated with mortality, and this all the other ones, the B cell receptors really don't show any significant association. We did some sex stratified analyses here as well, and these effects seem to be stronger in men as compared to women. The P for interaction was borderline at 0.06, so I don't know whether I can really say it is stronger in men, but looks impressive. So, this is my last slide. I think what we are doing here is to put another piece of the puzzle when we want to study the immune system and how it affects human health. We started off thinking these T cell repertoires are going to represent total antigenic exposure. But I think more that we look at this data, I think the explanation here is that a more diverse repertoire here represents a more robust immune function. And that, is consistent with negative correlations with age-related inflammation and higher levels of immune, so on and so forth. But also I think there is a second piece here that affects the diversity of the repertoires, which is these acute and repeated exposure. That's why, your positive associations with current smokers, CMV status, seropositive CMV status, which probably represents, recurrent exposure to CMV infection, elevates your repertoire, the number of clones that you detect here. What are next steps, I think? For HRS, or for other studies that may have RNA, maybe a deeper profiling of the T and B cell repertoires may be useful to confirm these findings. Typically with these targeted methods, you end up with between 8 to 10,000 T and B cell repertoires as compared to the hundreds that we have seen in this study. And then like looking to the future, I think many studies will move towards like single cell approaches because there you can actually get what we do with these bulk exposure, bulk sequencing approaches is really you get the alpha chain separately and beta chain separately. You don't really know which alpha and beta chains pair together. Single cell sequencing will allow you to get that. And if you get that, you can think about identifying specific antigens that they may be exposed to. But that's kind of fantasy at this point, but we'll get that at some point. So I just want to acknowledge, this whole idea started because I was giving an intra-departmental talk on what we were doing in HRS. And Mike Farrar is one of our immunologists, and he suggested I should try it and said he had no idea how to do it. So but he thought it would be a good idea. And, so Mike did that. Gokul is a student in my lab and really outstanding. This was all, without him, this work wouldn't have been possible. He figured out how to script, how to get Mixer working on a supercomputer, how not to break a supercomputer. And then, script all those 4,000 files to get it into a workable data set. And Ryan Martinez was a former fellow in our department and MD PhD fellow. He's currently an assistant professor. He's an immunologist as well, and he helped us interpret all the mixer outputs and how to get them, how to make sense of it. So I'll stop there and take any questions.
I have a question, and I don't know if this makes sense, but just to throw it out there, have you considered creating epigenetic biomarker proxies of these measures? And what would that, I mean, would it represent perhaps like a longer average estimate of these populace, you know exactly what to call them, prototypes, right? In the blood, would you get maybe a more stable measure that's linked more to sociodemographic factors? Or do you think that would not at all, as a biologist, not at all be?
I don't know. I think of these clonotypes as kind of a dynamic measure, not necessarily that they need to be very stable over time. But it's an area I haven't even thought about, so it's worth thinking, Epigenetics seems to be a valid measure of so many other things, so why not?
Or that it would be interesting if the point in time measure is different from the biomarker,
Yeah, it's a good point. Yes. We should at least think about how we would do it.
So we can't with this information identify antigens that were present, or can we get at least a vague idea? Could we even get like at the herpes virus level or?
No, so I mean, when we started the project, I thought that would be the goal. That would be cool. We'll identify CMV specific antigens or receptors. But I think, the two things. One is, we are using the pulse sequencing. We don't actually get the alpha beta receptor. But more importantly, I'm a little worried that, our reproducibility among the duplicates is only like 25%.
Right.
That suggests that, whatever sequence we are getting probably has too much error in it anyways. So I think we have to be a little bit careful about, getting to that next level, which is why I think, it is those single cell approaches, not that they're without error, but I think there are a few steps to take before we can reach to the level of antigens.
So, Bharat, one of the things I always deeply appreciate about you is you care a lot about measurement. You're doing a lot of work getting the measurement right before you, take it and run with it for prediction. But well, thinking about that, so you were talking about how to get better measurements. But is there an effort to try to take what you already have, and you looked at a few demographic things, but like HRS has, sometimes decades worth of information that could be used to predict these or later outcomes, and just how useful it is above and beyond, say, other biomarkers, or if it is.
No, that is work that we definitely are planning to do. This is as of last week.
Yeah.
So this is our first look at the data. I mean, we've had this data for a while. I was like terribly disappointed that it was negatively associated with age. So I kind of gave up on it for a little bit.
But it's associated, which
yeah, it is. I mean,
Inconsistent.
Inconsistent.
Negatively.
So I'm starting to think that it's the hypothesis that was probably not the data. So I think. You're exactly right. All these other variables we have in HRS over decades, starting to think about how they can be used to predict these type of long-term disease and treatment.
Should hold this. Okay, good morning, everyone. Thank you very much, Eileen, for the invitation. I'm very excited and honoured to be at this meeting today. I'm in LA, enjoy the environment here. So I'm going to talk about some work that we've been working on for a while to develop a new clock based on metabolomic data, which maybe is not as fiddly as the data we just heard, but it's almost fiddly, I would say. So this is work using a type of metabolomics. I'll try and explain all the different metabolomics a little bit, but using mass spectrometry, and it's in the Teal Eich and the UK table. So I didn't think I needed to put many introductory slides in, but of course now we're in an era of many different clocks. I know in HRS, I don't know how many clocks you've got now, but you've got an awful lot, mainly epigenetics, I think. But this is a lovely review a few years ago, sort of talking about the different omic levels and how they can contribute to making clocks and perhaps give us different or complementary information. And you can see in red, it talks about how there's now Nightingale, NMR, metabolomics, and we've now seen a bit of an explosion of some metabolomic clocks using this data. So obviously we all love clocks. I think for geroscience specifically, I think really we're interested in them. They might have more practical applications in the future, but I think at this point we're interested for what they can tell us scientifically about aging. So what are the determinants of the variation? You can use it as an endpoint. But also if you develop these multi-omic profiles, maybe we're finding some new biology underlying the aging process. So why metabolomics when we have all these other omics? So here's a sort of simple schematic of metabolomics, but metabolomics is really just measuring small molecules in the blood, small chemicals such as cholesterol. Eileen was doing the first metabolomics there, but we've expanded that now. But really we think of it as the final omic, so closest to the phenotype. So it's bringing together all these different biological processes, also the environment as well. And also it's giving us information about organs and tissues, not just cells that you happen to measure. So we and also we know that many of the hallmarks are very fundamentally linked with metabolism. So you can think of, for instance, the nutrient insulin growth signaling pathway is a fundamental metabolic pathway. So it should be very relevant. So that's why I'm interested in metabolomics for this. So I've been doing this for a few years now. So this was the first sort of attempt about 4 years ago. This was in an airwave study, so not really an older cohort, more a working age cohort. And here we use untargeted platforms. So we've got all these chemical features. Finally, we use different platforms. In the 4 platforms we use, we had about 28,000 data points, so quite big data in terms of the signals you get. And from this, it really showed that actually you could use metabolomic data and you could predict age very well. So average of about 0.86 correlation in the test set. So that was a good proof of principle. The age gap associated with risk factors. And you can do some sort of pathway analysis on these untargeted features at the pathway level. And the pathways coming out were certainly processes that occur in the mitochondria or related to intracellular signaling like tyrosine, tryptophan, metabolism. So this really gave us some idea that actually we were picking up aging biology. But actually this is not so useful if you want to extend this out and apply it to different people, because you have a list of untargeted features, and so these aren't quantified measures. So then we tried a different approach. This is using NMR. So you might have heard the Nightingale platform. So NMR, you get a smaller set of much better quantified, but actually even with this, you still need to do lots of normalization. This was really a mega study. So we were developing the clock in about 30,000 people in 10 different cohorts, and then trying to validate in the UK Biobank. Probably I should have done it the other way around, but you know, it started putting the states together and so on. But you know, and it works okay. But actually, with this NMR, you know what? I know people say you get 200 features with this set, but you really don't. You maybe get about 90 measures, and they're all very correlated. So we tried different models, and you can see these are the list of metabolites that you get in one of the models that we applied. So it's quite a small set. So I'm not sure you're really getting at aging pathways, but at least we know what the molecules are. And that way we can apply to different cohorts and do this large population scale epi. So this is what we're trying to do now, is sort of bring it together. So the aim of this is, I'm going to present one bit of work today, is to characterize the most important metabolites and classes associated with aging across the adult life, and to develop this multi-omic clock, which brings in a broad assessment of metabolites, and then do validation looking at phenotypes and mortality. So here is our study population. So we have 2 different cohorts. So we have Tilda, Catal's. Catal. I've given up. I've forgotten already. Completely lost. So Professor McCrory's cohorts, Tilda, which you know well. But then we also have another cohort, Airwave, you might not know so well. So it's an occupational cohort. So these are actually employees of the police forces from across Great Britain. So it's not a population study, but then this is the advantage of this study, is you have national representation, and you also have very good representation of the younger age range as well. So, you know, we wanted to do something across the life course before people are actually getting the diseases. So maybe we can argue this is better for looking at aging. So we have these 2 cohorts, almost exclusively white ethnicity. And in Tilda, we also did some additional sampling of cases, so we had the power to detect any associations with mortality. So altogether, this is about 4,000 samples from around 2,000 individuals. And we put them samples through this machine. This is the mass-spectrometer. Then we get all this raw data So actually, what you really get, you get the retention time, which is the time that the chemical comes off the column. So it gives you some information about polarity, that's liquid chromatography, and you get the mass to charge ratio, and this tells you about the exact mass. So you're getting 2 bits of information, and that's it, and no intensity. So then, like I say, we wanted to do something reproducible. So there was a big sort of process of annotation. And then from that, you would do a sort of targeted extraction and integration. And finally, we had a set of over 500 metabolites. So the product we end up with is a bit like Metabolon product, I would say. But I'll talk about some of the different advantages from our approach. And then we do the sort of standard machine learning. We use the ensemble lasso approach and then looked at the age gap with different phenotypes. So just to talk about the workflow, because I think it is important. So we start off with this messy data here. We have these repeat QC samples so we can do the quality control procedure, filter out sort of dodgy filters. And then we get a nice table of features, but we don't know what they are yet. So then there is a process and this is in collaboration with the National Phenome Centre, which is the centre we have at Imperial. They also already have some libraries, so they can, from previous work, so already we have a list of metabolites we can pull out, but we also do this extra untargeted analysis where we're looking across all these features. So we make sure we're not missing anything. So it's sort of a combination of the untargeted approach, but then also trying to quantify what we have. So just to give you a sort of idea, this is a UMAP. So it's a sort of non-linear principal component analysis to look at the metabolic space. And just to compare, these dots are different chemicals and the blue ones are chemicals. This is in 200 people in the airwave who have all the commercial providers as well. So really this is just to say, if you look at Nightingale, which is in yellow, you're only really assessing a little bit of the space, and most of these are these lipoproteins, cholesterol measures. But actually we have quite good overlap. So really we're measuring a similar thing to Metabolon. This is really what you can do now. And on one side you have the more small molecules, and on the other side you have the more lipids, more non-polar molecules. Gone the wrong way. So just to give an overview, so here we took this correlation map of metabolites, and we've overlaid associations from meta-analyses for Metabolon, for Nightingale, and also our National Phenome Centre data. So here you really get an overview. The colours are the correlations with age. If there's a shape, it passes Bonferroni correction. So just to give you an overview, you can see what are the most important species, really. So the sphingolipids are important, and also glycerophospholipids, there's an error there. But a really important one are these ones that go down, which are the sterol lipids, which is the steroid hormones, which is probably really important. And then all these acylcarnitines, huge differences there. And then at the top you have all your amino acids. So this is our overview. And then we did, like I say, this is what the raw data looks like. So this is almost 7,000 features. And you can see there's some very strong signals, as you might expect with age. So a p-value of 10 to the minus 40 at the top there. And so from this, we did, like I say, we extract the features, send them to the laboratory, then they do very laborious experiments. Hopefully they identify. So now we have added about 60 more metabolites that we know are very strongly related to aging. So that's the process. So now this is an interesting plot. So these are all our metabolites in the end. This is just plotting the age associations against the log hazard for mortality, adjusted for age. And actually, you can see that, as you might hypothesize, there is a significant correlation. So the things that are associated with age are also associated with your mortality risk, which I think is interesting for how you model a clock. Do you model it on age, or do you model it on mortality? And actually, they seem to be saying quite similar things here. But we modelled on age. So just to show you how we did it, we split up our data. So we had equal numbers from Airwave and Tilda from the baseline samples in our training, where we did this sort of bootstrap ensemble approach. So we're making lots of models and combining them together. And then we have validation samples, both baseline samples and follow-up samples. And we've also, the cases, because we have over sampled cases, but we've kept, our training is still representative of Tilda, so we have 13% of people will die during follow-up. But then because we want it to be very non-biased, we have a very separate sample for doing case control analysis for mortality in the validation samples. So this is how the plots look, and actually these are all validation samples, and it's, I think, on a par with most of the other clocks. So we have a correlation of 0.9, mean absolute error about 5 years, which is not so bad. We also plot the follow-up samples, so it seems to work, predict the longitudinal samples. They sit on the baseline samples in terms of prediction. And if we colour the age gap, which we calculate with the Horvath method, we're not calling it acceleration, even though it's the same formulation as age acceleration, but I think lots of people don't like that. But age gap is safer. And you can see there's an age gap going from -10, -20 at the extremes to 10 years plus. If we look longitudinally, so remember we have repeat samples, so we can measure the repeat. You do see everyone gets older metabolomically, as you might hope. I mean, there does seem to be an under-prediction, but generally the distribution around the follow-up samples is as you expect. So this is really important to say we're looking at things causally related to aging. I think this other plot is very interesting. This is plotting the metabolomic age gap over 2 time points. And it's remarkably consistent, even in the younger population, which is in red, and so these are middle-aged people, and in Tilda, who are in green, so it's about a 5-year difference. People generally have the same age gap between 5 years. So I'm not sure what that tells us for how well we can alter biological age, or whether it happens much earlier and then you're stuck on that trajectory. But it's an interesting phenomenon. So this is trying to describe, so remember this is bootstrap, so it's actually 500 models. So these ones funneling off at the end are the most important features that come up in all the models. And then you can see the contribution on the y-axis on the average coefficient. So it's quite a nice broad range of different features. So here's a table. I don't expect you to read this table, but these are the top, the ones that really come up in all the models, but there are many other metabolites contributing to these models. And you can see we have lots of acylcarnitines. You know, but actually the longer chains ones seem to associate positively with aging and the shorter ones negatively. So there's a sort of balance in this metabolic pathway. But also niacinamide, tryptophan, which has come up before, which seems to be very important. And there's some interesting new things that are coming up with this untargeted approach. So we have a glycan, so this 6C lactose, so it's not just your name. I struggle with it. It's the name. But this is a glycan, which is a sort of sugar attachment to sialic acid, which has been linked with the brain, bone health, and steroid hormones, bile acids, sphingomyelins, which are important for the membrane. So actually, we're getting quite a broad range of different molecules coming up, and they all seem quite interesting in terms of their biology.
Just say the positive versus the negative correlation coefficient, which one would be?
So the mean coefficient, if it's positive, it goes up with age. But also remember, this is a multivariate model, so it might be that there's a sort of correlated feature. So it's broadly correct, but you shouldn't interpret it exactly. That compound goes up that amount because there's correlation with other features. So yeah, an interesting range of compounds from a broad range of different classes. So this is the mortality analysis. So if we look at the age gap, this is a sort of case control analysis. Per year, we have an estimate of about a 7% increase in mortality. And this is very robust, really, for a range of different risk factors. And also, if we look at this predictive performance overall, but remember this is a matched case control, so the numbers aren't quite what you might expect in the real world setting. But you can see the metabolomic age gap is a similar effect on your mortality risk to what I would say would be the more clinical risk factors such as BMI, hypertension, diabetes, smoking and alcohol. And there does seem to be a gain on top of that. So it seems to be, could be important in a clinical setting maybe. And if we look at frailty and cognition, we see associations with the Freed frailty score. So about a 2% increase in the Freed score per year. And also the same with the MOCA cognitive assessment. And if you can also see this if you sort of use the cutoffs in the literature for mild cognitive impairment, there's a sort of 4% increased risk of maybe what might be screened defined as mild cognitive impairment per year. Grip strength are in the right direction, so it's not significant. And just looking more broadly across the different phenotypes with this metabolomic age gap, generally it is, as you might expect, it associates with more medications, greater depression. There's a sort of borderline association with menopause. And then we have various different cognitive measures that are presented MOCA that we see a consistent pattern with the different measures. BMI shows with an increase, and we also have increases in CRP, which are pretty significant, and also with some of the kidney markers, as you might expect, and the glycated hemoglobin. HDL is going in an interesting direction, but obviously this is part of the clock. But as we see with many other of these clocks, it's not really related to DNA methylation age or telomere length, so it seems to be telling us something quite different from these measures, which is interesting thinking about a little bit.
Wait, that said it was more about?
Yeah. So, sorry, I'm going a bit quick. So if we look at the bottom, which is MH, and you can see Horvath, I mean, none of these are significant. So Horvath, maybe it's a negative correlation. This is adjusted for age and telomere. So they're not really related at all. And if we look at the risk factors here, we've got airwave and tilde separated, obesity, hypertension, diabetes, depression. These are all consistently associated with increased metabolic aging. Alcohol use and smoking is quite inconsistent, I would say. And I think you see this quite a lot with these first generation clocks. And I think we need to think about what this means a little bit, actually. Why really associated, instead of thinking, oh, well, we need to make a new clock that is associated. And physical activity looks quite consistent. Education is as you would expect in Tilda. In Airwave, and remember these are police officers where education is not so important. My son wants to be a police officer, so I now need to be nice and that's please. And we see a different direction. So just to finish, we're also working on many other clocks. So one thing we're doing is applying these new somologic proteomic clocks in the EPIC cohort. And so this is EPIC, they're a bit younger, they're European wide and it's a longer follow-up, so you have to take this with a pinch of salt. But generally the different clocks out there, and there are 5 chronological age clocks out there, they're all having per standard deviation about 1.4. Well, 1.3 is 1.4, which is pretty similar to our metabolomic age estimate, and also very similar. This is some work that Em helped us with recently, so you should read it, but this is comparing different epigenetic clocks. And again, the first generation are all around the same. So it's working pretty similarly. This is quite, I just draw your attention to this. This is something we're doing where we're combining different trained populations and averaging out the clock. And actually you see a much stronger association with health outcomes. So I think this is telling us something about the importance of the reference population. Maybe this could be a way to improve the relevance of these clocks. Just brief rounding up, because I know we're already in coffee. So we've developed this clock and it works with frailty, cognition, mortality. And we think this clock is important because it's combining this broad metabolite coverage, this untargeted approach, but also this cross-cohort capability. So, you know, I didn't mention, I meant to talk about that we run these long-term standards so we can harmonize across cohorts. I'm going to talk about that, but that's one of the advantages we have with running it ourselves. And also it's an open science platform, I should have said that as well. You know, you can go and look at all the protocols, which you can't if you send your samples of the tabulon and everything. So it seems quite promising. Future work, we're going to integrate genetic data, proteomic data, so we have a bit more idea of what these metabolites mean and the pathways they are, and we can apply some causal methods. Finally, I work a lot with a cohort in London, which is a more smaller cohort, but it's very deeply phenotyped, cognitively healthy, and we're looking at progression to dementia. So this question of how biological aging matches to dementia pathology or not, we can explore here. And we're going to do a simultaneous quantification and profiling method, which hopefully will allow us to come up with a small panel of quantified metabolomic clock, which hopefully can be run much more widely. So yeah, thank you everyone. This is Chung Ho, who did pretty much everything here. And the guys at the National Phenome Centre, and of course our colleagues at Trinity College Dublin. We're very grateful for the wonderful samples to deal with. Thank you.
A broader question, more from naivety about metabolomics. I mean, there's definitely something very tantalizing about getting kind of to the closer biological substrate of phenotypes, but then you also worry about the potential variability and whether things like circadian rhythm, fasting, non-fasting status, how much that makes a difference. And I'm just curious as to whether there have been a lot of studies.
It makes an enormous difference. Yeah. So this is the you know, why epigenetics is great and metabolomics is challenging. And it makes an enormous difference. So, you know, what we try and do, partly by bringing in cohorts and looking for concordant associations across cohorts, is, you know, you take it, if you see it consistently and you see it with a long-term outcome like age, you can be surer that these are reflecting aging and they're not related to these things. But really, obviously, this is throwing in a huge amount of noise. Diet and climate there. So you try and control things, but it's probably making the signal weaker. I think it's to say.
Well, it goes back to what you said earlier today at breakfast. You said, these things aren't ready for individual usage, much of it. And that's where you're going to get that kind of variability, that they may be good for populations and saying something, but they're still not ready to.
I would like it to be successful for able to run in people and be used in a clinic, and that's part of why we're going down this route of
path.
quantification and standardizing.
And it's possible that your effects might be bigger too, right? If you're able to remove all that noise and actually be able to characterize it.
Right.
Yeah.
That's what I think. Yeah.
You're getting the start of where you're going to go.
Unless the noise is.
So thank you for the talk. I actually was going to say the exact same thing but that's actually what I find exciting about metabolomics, right? Is that it's unlike epigenetics, which changes either not at all or very, very small amounts over a very long period of time. It's good for long-term prediction. I like this is like could be a faster turnaround. And that one of the things that I kept thinking of, and then you showed the kind of the longitudinal similarity between is by training on age, by training on these things, if you've actually removed a lot of the noise, but you also removed some of the signal that we're actually the most interested in, which is the short-term changes. And that's why it's potentially, if it's the same at 2 time points, it could actually be something that's really early in life or genetic.
Yeah.
And then, I almost worry that it's lost some of that exciting, time-sensitive component.
Yeah, and also this could be a criticism of the algorithms we use as well. This is a criticism of lasso and penalized regression, that we're looking for things that don't change.
Right.
But yeah, no, you're right. But I think for this particular study design, we're trying to characterize these long-term changes. But no, you're right. There could be different ways to do this, that we might see some of these and maybe make it more sensitive.
Oliver, I think that was amazing work. First time I've seen it since you presented it last year. Could you comment a bit on the use of fasting versus non-fasted samples, and also the other potential confound, which I think we discussed before, was if you're taking medications like statins, or how it affects these estimates?
Yeah, this is going to be something that the reviewer's going to say. Why are these not fasted samples? And I don't think you had fasted samples.
No, that's not what I want. Yeah, these are not.
So, I guess the other argument could be, well, maybe in a population, if this is going to be useful, fasting samples is not so common. It works. But, it certainly, I think, would work a lot better if it was in fasted samples. This goes back to the noise issue. With statins, we discussed a lot of things about doing this, like should we residualize across the whole metabolite data set for statins? It's slightly problematic, but what we've done in the end is just adjusted all our associations for statin, and statin use isn't related really to the age gap measure, so that's quite promising. But yeah, that is another question that is going to come up. So I need to be practicing a bit. Yeah.
Very exciting talk. It's amazing that you have such great accuracy with metabolomic work.
Yeah.
I wonder if you tried subdividing your model into organ-specific blocks.
You from Bangladesh?
Exactly.
I was reading Ludwig, the paper in Nature.
Yeah. I wonder if you tried similar approach.
Well, this is why I'm excited to link to the proteomics, because metabolites obviously telling us something very interesting, but they're quite non-specific. They could come from many different things.
Yeah.
So I'm hoping with bringing in the proteomic data, you can really try and subdivide the metabolome a bit more. I'm not sure how I'd do that on its own. Short of looking at databases and so on, but like I say, most of these can be measured in different places. Some we know, like we know a lot of made in the liver, or a lot of reflecting in the function of the kidney. Yeah, I'm not sure. Sorry, yeah.
Yeah, maybe do you see some naturally occurring angiotypes in metabolomic level? I mean, clusters of metabolites that always change kind of together. And maybe they are reflecting some particular system
Yeah.
in the organism.
I'm not sure we have gone down this route yet. At the moment, we've been trying to annotate and get the data set together and put it in this model. But actually, that's another very good idea to do, to go down more in depth in the metabolite rather than reducing everything to one score, which is what is quite appealing, but is very reductive.
We're going to get started session 2 on neurobiomarkers and cognitive aging. First speaker is Teal Eich. Is that correct? Yeah. Neuroendocrine markers of cognitive aging.
Hi, everyone. So I just want to preface this by saying that I'm an experimental cognitive neuroscientist, so this is going to be maybe a little bit different than other talks in this session. But my background is primarily in understanding memory changes and deficits in aging, and then in particular how sex differences might affect prevalence of Alzheimer's, for example, the overrepresentation of women, and understanding why that is, when that happens, and timing hypotheses surrounding that. Okay, so this graph basically consists of data from a commonly used neuropsychological test, the DigitSpan test, which assesses memory capacity and is often used in the diagnosis of Alzheimer's. Can you hear me? Okay, especially yeah. And mild cognitive impairment. So in the test, the participant has to repeat in kind of a forward or backwards order, increasingly large set sizes of information, and they have to do this until they essentially fail out. And then you get a metric of how much their memory capacity is. And about 20 years ago now, Lynn Hasher at University of Toronto argued that, in fact, what led to the deficits that we see in aging and older adults, which are really, you almost always see these age-related capacity deficits in older adults relative to younger adults, wasn't specifically memory capacity, but instead due to older adults failing to reduce inference or interference from previous trials. So essentially, they don't drop the information from the previous trial in the set, and so they have more information that's clouding up search, and therefore this is what's leading to it. And she tested this in a really simple way. She just started at the larger set size. So, and what she found when she did that was that you can essentially rescue these memory deficits. So if you don't have all this competing information at the beginning, you can essentially get rid of, to a large extent, these kind of capacity issues with memory. And so I've been interested in this and how exactly do you reduce interference? And it's thought to be, we call this inhibition, right? So cognitive inhibition. And it's thought to be a mechanism that actively acts upon memory representations, again, or traces to reduce their strength. And it's often considered an executive function that enables us to resist interference from task or relevant information in the service of goals. And so when I've done work looking at how brain function or brain integrity is related to this ability to essentially resolve this interference and reduce or to use inhibitory processes to guide memory procedures. And I've done this in a number of different ways. So for example, we can use this really simple task called the directed forgetting task. And in this task, we present participants with really simple words or images, and we just tell them to look at them and then to either remember them or to forget them for a later memory test. And then on the test, we probe items that they were supposed to remember, like word paper, ones that they were supposed to have forgotten, like the word crown, and then novel items that weren't shown at all. And what we're going to do is look at the difference between performance on these two types of trials, the ones that we're supposed to remember versus the ones that we're supposed to forget, which are equally prevalent and require the same response. So we try to eliminate any kind of response biases and saying yes or no across these. And what we find and what we've replicated is that essentially younger adults are better able to drop. So when they're explicitly told to drop information and to not remember stuff, they cannot remember stuff. Don't use this in classes. But the older adults, it seems, hang on to this. And so they actually recall to a greater extent those to-be-forgotten items, which maybe is not a bad thing, but in this case is considered a failure of inhibitory processes. And what we found is that this kind of failure to inhibit these items is associated with thickness, cortical thickness, in particular brain areas, including the inferior frontal gyrus. And we've done this with another task as well. So this kind of probes this same idea slightly differently. So in this task, we give participants, again, words. We use words a lot. They're all rated and normed and everything for use in the English language and whatnot. So we give them 4 words, 2 in red, 2 in blue. We've tried this with 6 words. We've tried this with 2 words. We've tried this with very different things. And then after they're holding them in working memory, we tell them to either remember the red ones or the blue ones. We've done this with telling them to forget the red ones and the blue ones as well. And then again, after another probe, we test them on an item level. So rather than at the end of all these trials, we test them immediately. And again, we present them with ones that they were supposed to have remembered. So this was an item that was printed in blue, and they were supposed to remember the blue ones. Ones that they were supposed to have forgotten. So this was, or supposed to have inhibited. So they had to encode this word because they don't know which color they're supposed to be remembering. So they in theory have to encode all of them as well as their colors for the memory test. But they should have dropped these items and then ones that weren't present at all. And here we're going to test this slightly differently because now we're going to look at negative responses. So how good if people are responding no to items that either weren't present, the control items that should have no familiarity, or the items that should have been dropped or inhibited from memory. And we found the same type of the direction of the scale because we're looking at positive versus negative flips. But essentially we find the same thing. So the older adults seem to not be able to drop these items from memory, which is then impacting their memory. But it's an inability to filter out these items. They actually perform equivalently on the to be remembered and the valid items. And again, here we found that cortical thickness in the same area is predicting this ability. Interestingly, when I came here, there's a lot of interest in sex differences. And I had started looking at this, and when we dissociated, I ran the study in a larger sample. And what we found was that we just dichotomized by sex here. We found that, in fact, this result was driven almost entirely by the older women. So when you do comparisons across the old and young, you get age-related differences. But again, this was because the older women seem to be hanging onto and recalling this information inappropriately in the context of this experiment, at least. So this prefrontal region in the IFG seems to be important, at least in enabling this inhibitory control over the contents of memory. So how exactly does it do this? So Michael Anderson's group, he's a cognitive neuroscientist at UC London. What? Oxford, maybe. He found that memory suppression essentially engages this distinct frontohippocampal pathway that includes the IFG, which we found again to be this area that showed increased reduced cortical thickness. This was in a longitudinal study and related to these inhibitory processes. He also showed that hippocampal GABA concentration moderated this hippocampal PFC pathway, such that participants in his study, who were younger participants in this case, with lower hippocampal GABA levels, had impaired mnemonic control relative to those with higher hippocampal GABA concentrations. And so the lower hippocampal GABA participants in this model were essentially unable to exert this neuronal inhibitory control over hippocampal activity, which led them to fail to suppress items that they wanted to suppress. So what is GABA? So GABA is the main inhibitory neurotransmitter in the mammalian brain. And GABAergic interneurons help maintain homeostasis between excitation and inhibition by inhibiting and damping down neuronal firing. And a long history of animal work in this area has showed that GABAergic function is really critically involved in cognition and memory. So where there's dysregulation in the GABAergic system, one consequence of this is that you get hippocampal hyperactivity. So you aren't able to damp down the neuronal activity, and the result of this is too much neuronal activity in the hippocampus, and also impaired memory. And this paper in 2001 from Lee and colleagues showed using an AD transgenic mouse model who had reduced GABAergic receptors. And what they found was that this AD transgenic mouse model had reduced GABAergic receptors, hyperactive hippocampi, and this can be seen by comparing essentially the magnitude of the firing in the top and bottom rows of the graph on the left, and impaired memory. And interestingly, all of this preceded amyloid-related pathology. However, giving Boswell, which is a GABA agonist, rescued the hyperactivity. So this is in this red circle, and partially restored memory in these mice. So what about in people, right? So these are animal models. So hyperactivity of the hippocampus has been associated for quite some time now and is sometimes considered an alternative biomarker, brain-based biomarker of AD risk in people. And so what I'm showing you on the left is just different, or I'm going to show you 3 slides that are different ways of grouping people as a function of hyperactivity in different ways of categorizing AD risk. So for example, here what I'm showing you is people with and without an ApoE4 allele. And what you see here is that those with the E4 allele have greater magnitude of hyperactivity or greater magnitude of activity in general than those who don't have this risk. Here's another way of doing it. You just take people who are research clinically diagnosed as controls, people with MCI or Alzheimer's or AD patients. And again, you see here the MCIs have higher level of hippocampal activity than do the controls. The ADs by this time, you're wondering, probably have just fallen off and they're not having a functional, correct functional response to this. And then this one I think is actually one of the coolest ones, which is from a longitudinal study where they just looked in the anterior and the posterior hippocampus activity at people who did and did not drop out of a study, of a longitudinal large cohort study. And so if you go back and look at their baseline, time 1 or the earliest time point you have of MRI activity, you can see that the ones who drop out, even at the baseline visit before they've dropped out, show higher hippocampal activity than those who do not. And so hippocampal activity, hyperactivity reduction in MCI has been studied and has been led really by Michaela Gallagher at Hopkins and she uses a task developed by Craig Stark at UCI called the mnemonic similarity task. And so in this task, it's a really simple task and you're just shown a series of pictures of common objects. Like this apple, and you just have to make a response that's bold. So have you seen it before? Identically similar. So is it similar to, but not identical to a picture you've seen before, or new? There's various different ways you can do that with bold or new. They've done it in a gajillion different ways to really, and this is kind of a really good, consistent way of looking at this. And so what they're interested in here is this spread during these lure items. So this slotted spoon is similar to, but not identical to, this one that they've seen before. And in theory, this represents when you get this lure item, if you incorrectly say that it's old, this represents a failure to have pattern separated or have episodically separated out the details from this episodic memory that say that slotted spoon is similar to, but not identical to that other one. So you have a familiarity with it, which is correct, but not that episodic memory, which of course is what is a hallmark clinical symptom in Alzheimer's disease, is loss of that. And what they found was that giving levetiracetam, which may be related to GABA, although that's arguable, and the pharmacologist can speak more about that in the room. So they found in their first phase 2 trial that they had took AMCI patients, so amnestic mild cognitive impairment patients, and that giving this this drug that essentially is Keppra, that damps down neuronal firing in the hippocampus, also was associated with somewhat improved memory in these patients. I think that the larger phase trial has, at this point, not succeeded, but they're still doing a lot of their publishing daily on this right now. But they didn't measure in these studies, they hadn't measured hippocampal GABA itself. to see if this is actually the source of this hyperactivity, that then we can try to target hippocampal GABA more directly. And so just to back up a little bit, so what does GABA do across the lifespan? Why would we expect it to be changing? This is a systematic analysis from Voorhees and colleagues that showed that essentially GABA is relatively stable. This is all mostly prefrontal GABA concentration measured because it's very difficult to get GABA measurements in the hippocampus. So what they find is that, you know, it's relatively stable across these cohorts until this kind of inflection point at midlife, right around in the 50s, when then it starts to decline. And so I was interested in this, and so did a very small pilot study when I was a postdoc, just to ask a really, really simple question: is hippocampal GABA related to memory in aging in older adults? And basically we found that it was, but it was again driven by the women. So all of these women were postmenopausal around ages 55 to 70. And essentially the line was relatively flat for men. So this didn't impact them, but for every decrease in 1 standard deviation decrease in GABA concentration in the hippocampus, there was a memory accuracy decrease of 11% in the women. We also found, we have unpublished data showing that the GABA by sex interaction was modulated by cortical volume in these parahippocampal regions in the women. So why women, right? If it's true, this is the running hypothesis. Estrogen is neuroprotective, so it increases synaptic plasticity, improves hippocampal memory. So on the left, for example, is data from a hippocampal-based learning test. And the E here, it's a little, sorry, it's so light, but the E is estradiol-treated overoptimized rats who learned more quickly than, i.e. fewer trials to criterion on this test than in oil-treated rats. And on the right, this is in reproductive age women. Serum estrogen levels, which rise mid-cycle around ovulation, this correlates with verbal episodic memory. So the black lines here, the other lines are women who are on hormonal birth control. And there are very, 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 very few studies of this. But estrogen has been shown in some studies to impact GABAergic function. So on the left here, I'm going to show you now, is GABA measured in the prefrontal cortex in women with and without either anxiety or depression across the menopausal transition, right? So this is the beginning is before and follow-up is after the menopausal transition. And basically what this shows is that there's a steeper rate of decline in those with the psychiatric features, but this is also occurring, this drop in GABA are essentially occurring in prefrontal cortex, at least, is occurring in all women across this transition. And here on the right shows, again, prefrontal cortex GABA in reproductive age women versus perimenopausal women. So this is the time leading up to menopause when FSH starts to increase and estradiol starts to decrease. And again, the perimenopausal women who are closer to menopause show lower levels of GABA, of cerebral GABA. And, you know, animal models, which of course require caution because mice and other animals cannot go through menopause. One early study reported that overoptimized rats treated with estradiol showed increased hippocampal GABAergic neurons as well. That's that guy. And so this kind of background has essentially led me and my lab to what we're doing now, which is a study that is trying to kind of piece together these different parts and potentially get a mechanism to understand why, you know, we have this overrepresentation of women, and maybe it's something having to do with GABAergic function during the menopausal transition or after it that's then impacting AD risk and increasing these prevalence rates. And so we're looking at a bunch of different things, and basically the study design is that we bring in people from Mara's Healthy Minds pool here at USC, as well as GeneMatch, and we are going to test about 400 people. We're about halfway done right now. About 30 of them are MCI, although more of them turn out to be relatively not so cognitively intact these days than I was expecting. And we've dropped the age range to include 45-year-olds. And then in person, they come in and they donate blood, urine, and now, because of my graduate student Doak Young, nails, where we can get a chronic measure of stress. They do a neuropsychological battery, do a really detailed medical history, complete magnetic spectroscopy, resonance spectroscopy, which is then processed by Minnesota from their pipelines. And they do an fMRI task where they do that same picture recognition task where they make old, similar, new decisions, so we can look at hippocampal hyperactivity or hippocampal activity as a function of different ways of classifying these people. And then at home, we make them do a whole bunch of questionnaires and stuff that we're trying to look at. So far, we have 174 people who have completed this. About 30% of them have an ApoE allele, either E44 or E43, 3-4. It's relatively good demographics on it. And this is, there's a lot of data, and I literally got some of this like really, really recently. So this is not, the hormone data especially, which just came back from Oregon, is not clean yet. And it's also very variable, and we need to look at things like medication use, because we exclude for any kind of medications that would target the GABAergic system, or we have to look at all of these things, and we haven't quite done that yet. But just as a sneak peek, so this is hormone levels, and what we see is, as one would expect, so biological sex results in decreases in the females in estradiol and progesterone, which is to be expected. Again, these women are all 50 plus, and we have about 96% of them are postmenopausal. And again, we're trying to drop down the age because of that, so that we get more variability in those to look at timing hypothesis during the menopausal transition rather than after it. So for GABA and glutamate, what we find is that GABA decreases with age in the women only, but not the men. And that glutamate decreases in the men only, but not the women so far. And again, this is really preliminary data, but it supports part of the hypothesis. And when we look at, this is in the women only. So when we look at hippocampal GABA by memory, and again, this is people who respond. This is the probability of responding incorrectly. So responding that an item that is similar to one that one has previously seen, they respond that it's old. So they have a failed ability to pattern separate out and have an episodic memory of the new versus the similar item. So here, higher probability is worse. Reduced GABA or less hippocampal GABA is associated with both being older, having less estrogen, that's a little bit fuzzier, and making more episodic memory errors. So this is not quite significant at this point with a relatively small sample, but it's going in the right direction. And then finally, just because we've had this data longer and so been looking at it, this is just women only to say that this experimental memory task is again an experimental memory task, but we have a standard neuropsych battery which shows pretty consistently normal results across our entire sample, as well as in women only. So in that you get these memory, speed, executive function deficits, and these increases in language or semantic memory. And so thank you.
About 3 minutes here.
So thank you for this. I feel like I know about the issues around fMRI and I have worked in that. I don't know all about hippocampal GABA. Could you just tell me a little bit more about how that's collected and liabilities and things like that?
Yeah, it's really hard.
Yeah.
It's really hard because, especially with age. So we brought in, there's only a couple of people who do this. Most of them are at Hopkins. And so we brought them in, as well as Minnesota. They have the good pipelines, and we did a lot of piloting on it. We do get pretty good quality control measures in the moment, looking at what the spectra should actually look like. And we the hippocampus is actually not so hard to find anatomically. For example, we tried to run DLPFC, the frontal region that everybody uses, and frankly, our data was a complete mess. So I don't know how people do it because the landmarks are much harder, at least they were for me, to find. But we do a lot of quality control on it. That said, most people that I've talked to have to exclude like 60 to 70% of their data. We're at about 30%. Even with good line widths, good quality control, perfect spectra. So I don't know, it's hard. It doesn't take a super long time to run. It's like 13 minutes a scan, but it's still, it's not easy data to work with.
Just as a quick follow-up, do you ever look at like the people who don't have it and how they predict anything?
So I haven't yet, but we can, because we have people who are, 6 standard deviations off on this. And I think it's hippocampus specifically because of course we do a control region, which I didn't show you in the posterior visual cortex. And that data is much cleaner. So, and that would be expected because you're not going to get pathology there. So it might be some interaction with just age-related pathology in the hippocampus. And we can do tissue correction and all different measures for that. But the visual cortex is much cleaner and easier to work with data.
Do you collect data on sort of age at menarche, breastfeeding, and child history? And do they influence the?
So we haven't. We have to stay for it. We had to really build a big comprehensive women's health survey. Because the Menopause Society has a pretty good survey, but it doesn't quite tap into all of it that we want. So we do have all of that, and we have predictions about age of menopause, and we include women here with hysterectomy and fewer than 2 ovaries. So we're gonna have to, again, these are all like an HTUs, right? Because the HTU, that's I didn't show the estradiol data, maybe mainly because we haven't gone through it yet to say who hasn't used, and there were a large portion of our population who has.
So the findings are great so far. The men in the glycogen, was this predicted?
Yeah.
Okay. And do you have any? Oh, I see. Okay.
Yeah, no, I don't. I would love feedback on if no. But it's sometimes people take a ratio of GABA to glutamate, and and in theory it should. So yeah, I don't know. I have to think about that one. And they don't seem to. They seem to be more correlated and both decreasing in the visual cortex. So it's hippocampal specific in this preliminary. Yeah.
A question about the premise to some of the behavioral measures that you were talking about at the beginning. The interference that you started with, and then I think it was Gallagher's pattern separation. Both of those were failures with age. But if you flip it, they also sound a lot like increased gist.
Right.
It's also
Definitely.
kind of
Right. So it's definitely a shift away from episodic towards gist, which may be, which maybe is adaptive, right? Like maybe it's not, I frame it as like it's bad, but maybe it's not bad. Maybe it's just a different strategy. So we're definitely thinking about it in terms of a detail to shift switch and
what's gist?
Just like you don't remember the episodic details again. You go towards having the gist, the schema, of what you remember that it was a spoon. You just don't remember it was that spoon.
All right.
Okay, thank you.
Sounds good. Talking to us about pathways linking the dramatic changes seen in autonomic activity to changes in amyloid beta levels from ages 20 to 60.
Great, and Steve is one of the collaborators on this, actually. So really, thank you for inviting me. It's great to be here today. What I want to talk about is what happens between the age of 20 and 60 that increases the risk of Alzheimer's disease. So we all know age is, one of the biggest predictors of Alzheimer's disease. And there's probably lots of reasons, but if we think about what has been guiding Alzheimer's disease research, of course, it's the amyloid hypothesis. And this has been around for many years. It's been very influential, somewhat controversial. It's had its ups and downs, its successes, its non-successes, but the genetic research really supports that there is something about amyloid beta that it seems to be a trigger for the disease. So those autosomal dominant cases where there's some sort of mutation that predicts with near certainty that you'll get Alzheimer's disease, all influence pathways involving production of amyloid beta and where the amyloid precursor protein gets cleaved. And so in terms of the Alzheimer's, the amyloid hypothesis, the idea is that there's two high levels of amyloid beta peptide. So amyloid beta peptide is a normal byproduct of cellular metabolism in the brain of neuronal activity. And it also is something that can lead to these amyloid plaques. So it's the main ingredient in the amyloid beta plaques that are the main symptom of Alzheimer's disease. So for years, we've used cerebrospinal fluid as a way to measure this in people, and it's a little counterintuitive. So those of you who are not Alzheimer's researchers, this is pretty confusing, but it is a very reliable signal, which is that older people who have lower CSF amyloid beta are at higher risk of Alzheimer's disease, more likely to have the disease. Why is that? It seems to be because the amyloid beta peptide is no longer being cleared from the brain to the cerebrospinal fluid, and so they have less of it in CSF. And you can see it in people and animal models that there is this trajectory where you see, yes, you can see my mouse, you see an increase in cerebrospinal fluid amyloid beta 42 through age 20 to age 60-ish. And then what you're seeing is what researchers are more familiar with looking at. So, usually Alzheimer's researchers are studying old people or people with Alzheimer's disease. And in that population, having low amyloid beta 42 is a bad thing in CSF. That's like one of the primary indicators that you might have the disease in your brain. But this side is something that just hasn't been focused on. And in fact, these data are pretty unique. They're from a harmonized database with 475 individuals with longitudinal CSF. And you can imagine it's very rare data, right? Because who's going to study people between 20 and 60 and give them, ask them to undergo a lumbar puncture? But these were, I think, a lot of them were controls for the autosomal dominant, cases of Alzheimer's disease who know that they have the gene, but they have family members who don't have the gene who also enroll in the study. So a lot of these were normal people enrolled for that reason. And you see this dramatic increase in cerebrospinal amyloid beta 42. And those that working in the Alzheimer's field actually find this really weird. It's not weird at all from the amyloid hypothesis perspective, because the hypothesis predicts that it's the level of amyloid beta that is the trigger for a plaque to start accumulating and for the disease progression to happen. In this sample, they had APOE4 allele status, and when they separated the cohort by this, you get this very nice picture here, which is that the peak in amyloid beta 42 in CSF is happening earlier in those with the risk allele, and of course, they're also getting plaque earlier. We don't have that data from the same people, but that's generally the case. Whereas those who don't have the APOE risk allele are continuing to show increases. So each of these lines, sorry if it's hard to follow, this here is the whole sample, and they took those 18 to 45 because they took a pretty broad range because they didn't have as many younger. In here, we're just looking at each of the age cohorts, and you can see they each increase across the years that they're being measured, except for this one anomalous cohort, but in general, you're seeing increases in CSF amyloid beta 42. And this peak corresponds with about when you start seeing amyloid plaque in healthy older adults in just post-mortem data where they're unselected cases. So we have this trajectory. What the question is, what's driving this? Why is amyloid beta 42 increasing so dramatically between age 20 and 60? And this is the question that I think might be related to another dramatic thing happening in this age cohort, which is that autonomic activity changes quite a bit, and it has not been appreciated how much it changes. So, we have two major branches of the autonomic nervous system, the sympathetic and the parasympathetic, and they can act in concert, but they also tend to take turns. And so in the daytime, when you're active, when you're doing things, sympathetic noradrenergic activity is higher. At nighttime, the parasympathetic, and we can say also vagus nerve, which is the primary transmission corridor for the parasympathetic system, is also more active. What happens in aging? You might think, if you were just naive to this, that what would happen in aging is that the fight or flight, the sympathetic system would fall apart, right? That's the active system. But in fact, it's the parasympathetic, the rest and digest system that crashes and burns as we're going through aging from age 20 to 60. So 8 million Fitbit users had high frequency heart rate variability assessed. It was about 80% lower in those age 60 than age 20. And that sort of circadian rhythm that I was showing you decreases quite a bit by age. So this is another cross-sectional study where they had over 900 people, and you can see those who were 18 to 24 have this very robust circadian rhythm for heart rate variability. Heart rate variability is the primary measure of the parasympathetic system that we use in people. And I could explain if you want in questions why that is. But this heart rate variability is quite high during the night, middle of the night, and low during the day. You can see this is dramatically decreasing even in one's 20s to one's 30s, 40s, and it sort of just flattens out. These are not even old people, just age 55 to 67 here. So, and one more set of data on this. There are some very nice longitudinal data. No, I can't find any longitudinal data on the age 20 to 40 sort of span, but this Whitehall study in the UK did start with age 44 to 49 cohort. And this is just crazy as well. I mean, 10 years, they decreased high frequency heart rate variability by over 50%, both men and women. And it decreases as well by decade for the older cohorts, but less so. I think it's just, it's already gone, right? So massive changes happening there. And at the same time, we have sympathetic hyperactivity as we age. So there's lots of ways to measure sympathetic activity. One is that you could put a needle in and measure the activity of your sympathetic nerves, and those nerves are hyperactive. You just measure the activity, they're firing more in older people. You can measure plasma noradrenaline, you can measure CSF noradrenaline. All of those go up with age as well. And we don't have the beautiful curves in any studies, but like for instance, in this study, plasma noradrenaline was much greater at night in older adults and somewhat greater during the day. So again, we've got that flattening of the curve, but here it's going up. So we have these two things going on. My hypothesis is that these are really related to each other in a causal way, and that these changes in the autonomic activity are leading to this greater production of amyloid beta. And there's, evidence in the literature that suggests this. We know that stress is associated with greater risk of Alzheimer's disease, and in animal models, we can exacerbate Alzheimer's disease pathology by stressing the animals. There's this one study that would have been the best study to be a participant in. You got randomized to go to a retreat and be on vacation, or randomized to go to a retreat and meditate. And they found changes in plasma amyloid beta levels. They found changes in both groups, interestingly. So it wasn't meditation. It seems to be something about relaxing and going on. So something, about physiology. And then we know that, and this has been well studied, that sleep every night reduces CSF amyloid beta, and you can increase CSF amyloid beta by depriving people of sleep just one night. So every night sleep does some good things. And in general, from this sort of picture, it seems like that states associated with higher parasympathetic or vagal activity reduce amyloid beta, whereas those associated with higher sympathetic or noradrenergic activity increase it. So this is the model that I think from the literature might explain this at a molecular level. So amyloid beta is produced by amyloid precursor protein being cleaved in a certain place. So amyloid precursor protein is this transmembrane protein. So this is a membrane of a cell indicated here. This protein spans this membrane, and it gets cleaved by either beta secretase or alpha secretase. If it's cleaved by beta secretase at this particular location and then subsequently cleaved by gamma secretase, you get these amyloid beta peptides. That's the amyloid producing pathway. And there are a couple of animal studies showing that stimulating noradrenergic receptors increases this pathway. On the other side, you can have the initial cleavage being right in the middle of where the amyloid beta peptide would be, and that prevents amyloid beta from being produced, and then you have subsequent cleavage by gamma secretase. And there's a lot of studies showing that there's cholinergic activity that biases towards that pathway. So these studies are both in animals and humans, and you know there's like the early drugs for Alzheimer's were cholinesterase inhibitors. Those increase acetylcholine, and they found in patients that they decreased amyloid beta levels in CSF. And this seems great, and you know, why haven't those drugs been used for prevention, which is the obvious place you would want to be targeting the production of amyloid beta? There's a problem, which is that the drugs have side effects, in particular gastrointestinal side effects. So they're you know, not something you'd want to give to a healthy person. But if we might be able to do something behaviorally that would actually stimulate these systems, that would be really terrific. And the cholinergic receptors, the muscarinic receptors, they are stimulated by vagus nerve activity, and they're stimulated in the brain. So could we devise something that might be able to reduce production of amyloid beta in healthy people? And currently, there is no such intervention that we know of in the field, other than what I'll tell you about in a minute that we're working on. So sleep seems like the obvious intervention, right? Because we know every night that it reduces amyloid beta, and there's lots of people working on this. So far, there have been no clinical trials with a randomized control where they've shown that you can improve sleep beyond what you get every night normally to reduce amyloid beta. They're, you know, probably the population you want to look at most are people with sleep disorders like sleep apnea, and they've been doing that. So sleep apnea, a great treatment for it is CPAP, and they have shown that there's a correlation. Even here, that wasn't significant. They didn't have a randomized trial, but they've seen that those patients for whom CPAP improves delta power during deep sleep, they also show decreases in CSF amyloid beta levels. So it looks like you might be getting some of this decreased production benefit. Exercise is, of course, something that's been looked at, a behavioral intervention that's been looked at in a number of trials, randomized trials, but it has lots of benefits, but it doesn't act on this amyloid beta pathway, as far as we can tell from the studies. So one approach that might work is to actually stimulate what's called the baroreflex, which is the blood pressure feedback system that we all have in our bodies, where we have mechanical receptors on our vessels that sense stretch, and when there's a lot of blood, they send signals to the brain to activate a few different feedback pathways. One is through the sympathetic system, and one is through the parasympathetic system. The sympathetic, one of the pathways is to reduce the constriction. So the sympathetic system is responsible for blood vessel constriction. And so if you block that, you're not going to have as high blood pressure. So that's one pathway that gets activated whenever the body senses that there's too much blood pressure. The other one is the vagus nerve signaling to the heart to slow down the beating of the heart. So your heart has its own pacemaker cells. It's interesting. I mean, it depends on age, but it would be about 100 beats per minute. You know that your resting heart rate's lower than that. It's because of tonic suppression by the parasympathetic vagus nerve to slow down your heart when you're resting. And when that mechanism is activated by these baroreflex feedback loops. And in the brain, the locus coeruleus is the primary source of noradrenaline, and stimulating the baroreflex actually decreases its activity in animals. Breathing is also interesting because when you inhale, the vagus nerve doesn't transmit signals as well as when you exhale. So that tonic resting brake on your heart is lifted every time you inhale. So if you are at rest and your heart rate is much lower than your pacemaker cells would have it beat, every time you breathe, you'll see a big difference. If you're active, if you're stressed, that's not going on. You don't have that parasympathetic suppression going on. You won't really see much effective breathing on heart rate. So these are traces over 4 minutes of heart rate in people just sitting and resting in our lab. And you can see there's various, sort of oscillations that vary by person. But what is quite striking is that all these people are showing this regular oscillation that is associated with each breath, an increase or a decrease. So if you were measuring someone and having them do paced breathing around a normal breathing pace, this would be around 4 seconds per breath, you'd get this very high peak in spectral frequency of the heart rate at the breathing pace. But you also see peaks at other lower frequencies. So for instance, this frequency at 0.1 hertz or around 10 seconds is associated with the baroreflex. So there's an oscillatory dynamic associated with that feedback that goes to the brain to then trigger this. So it's always responding to old news, which is why it's an oscillation. So interestingly, if you breathe, you can move your breathing around. You can't move the baroreflex oscillation around, but you can move your breathing around. And if you breathe at around this 10-second pace, you get very high oscillations in heart rate. Presumably, the idea has been because you're perturbing the signal with both the baroreflex and the breathing. So we see this is the same for participants. Now they're breathing to a pacer in the orange trace, and you see these very large oscillations. It might be like from 60 to 80 beats per minute. So a very large physiological effect. And these sort of manipulations of slow breathing while looking at your biofeedback of your heart rate has been used as a therapeutic method for participants. Usually they do this, 20, 40 minutes a day, and they come in the lab and the researchers identify what pace leads to the largest oscillation. Then they go home and they practice this. And meta-analyses now have found that this practice decreases stress and anxiety and, has and depression as well. So it has emotional benefits. And we were interested initially, we set up a clinical trial and got funding for it in order to look at emotion-related brain networks and how this practice might change those. And so we set up the intervention group to do this standard sort of biofeedback practice. And we told them that we were interested in how heart rate control might be one of the reasons that meditation is beneficial. And we told them that some meditative practices lead to large but smooth oscillations in heart rate. And that's true. We were very concerned about demand effects. And so we designed a active control that had the opposite goal, but got the same biofeedback. So their goal was to try to keep their heart rate steady. And they weren't given breathing instructions. They could breathe however they wanted, but they had the same biofeedback and they had the same amount of practice. And we told them that some meditative practices lead to a low and steady heart rate, which is also true. Meditative practices vary quite a bit and have a lot of different physiological effects. And we gave them laptops, we gave them ear sensors. So we had measures of every time they practiced, we had their trace of what their heart rate was. And we could see that the people who were practicing the slow breathing showed these large oscillations compared to their own resting state. Those in the trying to decrease oscillation group showed no increases relative to their own rest, which is good because usually if people are relaxing, they show increases. So we want the biggest differences between our two conditions. And we had a exploratory measure, which was to take blood and look at plasma amyloid beta. And this was for various reasons. We were interested in this pathway and how it might be related to the slow-paced breathing and the autonomic changes. And we found large effect size differences between the two groups in both older and younger adults. So the amyloid beta 42 and amyloid beta 40, both of them overall, we just look alone at the oscillation plus group, the ones that were trying to make their heart rate oscillate, they showed significant decreases across 5 weeks. They were doing this breathing practice a couple times a day up to, by the end, they were doing it 2 20-minute sessions. And we had really great compliance from people, in part because of our setup where we knew what they were practicing and we could send them targeted friendly reminders, just to make sure people didn't forget. But people like doing it. Our older adults did more than 100% of what we asked them to do.
Okay.
Wow.
Can they see in real time their heart rate while they're doing it?
Yeah, not almost real time. So it's like just a couple seconds delayed and they're seeing
So
that.
they're training themselves.
Exactly. Yep, exactly. So then the other interesting thing is that the group that was trying to keep their heart rate steady and also trying to be in a meditative state, they showed increases in plasma amyloid beta in both the older and younger adults. So we were very interested in this. We designed a second clinical trial where we really leaned into the breathing and trying to increase those oscillations as much as we could. And we designed the intervention so that it was adaptive, and over time it would go towards a breathing pace that led to the largest oscillation for that person. And what that ended up doing, it turned out that the field's assumption that breathing at around 10 seconds leads to the largest oscillation is not true. It's the slower you go, the larger the oscillations are, as long as you're comfortable. And so these people ended up breathing much more slowly than our previous trial, and we didn't see the plasma amyloid beta differences. So we think that it has to do with the baroreflex because of that pace issue. And we, in a third clinical trial, it supports that the 10-second pace does lead to this. In this trial, led by Karu Nishiro, we had 3 conditions. We were interested, as our cover story for the initial study was correct. We were really interested in how these different physiological aspects of meditation might be leading to different effects. And so in this study, we explicitly tested that. We had participants, we did a lot of pilot testing of what does mindful meditation do does it make people breathe slowly? For novices, it didn't, which we thought it would, but we told them these standard meditation instructions, pay attention to breath in your belly, feel the breath. In the novices, it didn't increase their slow breathing generally. So we took advantage of that and had one group do that, listen every day for 2 20-minute sessions for a week to this guided meditation of paying attention to their breath. The second group had the same exact recording, paying attention to their sensation of breath, but twice in the recording it said, now try and breathe in for a count of 5 and out for a count of 5. Pace your, it's 1, they paced it during auditorily, and then keep doing this, keep breathing slowly during your mindful meditation. So same mindful focus on breath, but one was explicitly told to breathe slowly and the other wasn't. And then we had a third group, which it's really lovely to be able to include this do nothing group. They wore the sensor and they could do whatever they wanted during that time that they wore the sensor every day. So they had to sit still. So the 3 groups, we have all their data from their home practices in terms of their heart rate. And you can see that the slow breathing condition was doing this slow breathing because their heart rate is oscillating at around 10 second, a little, centered around 11 second probably. And the mindfulness group doesn't look all that different from the no intervention group. Now for the plasma amyloid beta results, we see very similar results to that first study where the group that's doing the slow breathing showed decreases. The group that was meditating without slow breathing showed increases, and the group that was doing nothing showed no changes. So again, we're seeing these physiological effects. It's very interesting that mindfulness alone does something that is completely flipped by adding breathing to that. But in any case, what in summary we're seeing is that parasympathetic activity decreases dramatically in early and middle adulthood, while sympathetic activity increases. And we're suggesting that these autonomic changes in early and middle adulthood may be an important factor in influencing Alzheimer's disease risk, and that breathing at around the baroreflex frequency range significantly lowers plasma amyloid beta levels, whereas breathing at slower frequencies did not. And a very important next step for us is to measure CSF. All of this has been in plasma, and in plasma, one of the primary sources of amyloid beta is actually platelets. So Steve Cole's lab did some analyses of platelet factor 4 and p-selectin, and we found very strong correlations between change in plasma amyloid beta and those platelet markers. So platelets, before researchers had really good access to brain tissue, and they were using platelets as a model of amyloid beta production, and it has all the same machinery, that APP machinery that I told you about is all present in platelets and also in neurons. So my hypothesis is that it will act the same way in brain, in brain tissue, in neurons. The APP pathway will also be influenced in the same ways, but we don't know. So we have a proposal under review to collect CSF before and after one of these interventions and see what whether we see comparable effects in the brain. And of course, if so, we're also very interested in how long-term this might affect cognition. So very interested in your questions, comments. Thank you. Mitzi.
Yeah, so that was really interesting, and I found it very fascinating. I'd never seen that data before showing the increases in CSF beta 42.
Nobody has paid attention to this, and the authors of that paper expressed puzzlement.
I'm sure they did.
Yes.
I'm sure they did, yeah. And I think it's interesting because we don't have a lot of data, obviously, from people that are that young, but to my knowledge, like if you look at PET or if you look at autopsy, you see, you don't really see much at all, right, in terms of pathology, but certainly there's generally a trend, which makes me think about CSF flow dynamics, because certainly heart rate variability is linked to CSF flow dynamics. And the thought with, why the A beta 42 goes down when you have Alzheimer's disease is the fact that you're getting more 42 or 40 production, right? And so then it's aggregating in plaques and it's not being cleared out. So I'm wondering if as heart rate variability is increasing, you're getting more CSF flow dynamics, you're getting higher levels of A beta 42, but then as you start getting more 42 production in the brain and more of those oligomers, that you're actually then starting to have more of the plaques form, so you don't see this benefit. I think it's super interesting. I'd love to see data where you have.
And that, honestly, that's the reason we included these exploratory measures, was because I was very interested in lymphatic clearance. Mm-hmm. And there's lots of reason to think that this should be increasing lymphatic clearance. So lymphatic clearance is this waste clearance system in the brain that is in these peravascular spaces. Waste gets cleared out. So there's a flow dynamic, and it requires fluid to be flowing in the interstitial spaces between cells. And those spaces between cells shrink when you give, when in animal models, they give a cocktail of noradrenergic agonists. So basically noradrenaline swells up cells and shrinks the fluid spaces between them, which makes it more difficult to have this waste clearance system operate. So that's one reason. And then, of course, the other is that this whole waste clearance system is on the fluid filled spaces are on the outside of vessels in the brain, and vessels propel that fluid by the dynamics of the heart beating and breathing. And that's been shown in, sort of MRI CSF studies. So I had this whole hypothesis about waste clearance, and we see a hint of that in that the older adults in our first study show improved amyloid beta 42 to 40 ratio at a p.05 level. So the younger show no sign of it, which makes sense because they don't have waste clearance issues. So I think that probably the breathing can act through multiple pathways. And in the younger, what we're seeing in younger people is this quite robust change in amyloid beta levels, which I think is a production. Thoughts, yeah.
Really interesting.
I mean, I think your work is just fascinating if you're working in sort of health inequalities and intangible things like psychosocial stress to actually see the data going all the way through. It's amazing. But just coming back to the point we were just discussing now, I mean, I guess, do people know how CSF amyloid beta relates to brain burden in the younger population? I mean, it's probably not the same mechanism in the younger group and the older group.
So,
To this CSF.
So this sort of curve in these data with humans, we don't have the pathology data linked with it, but there are many animal models of rodents and amyloid, and across those studies, what they find is that the CSF increases in those CSF amyloid beta increases in those animals until right around or right before the point when they start getting the amyloid beta plaque. So I think, and it fits with just what we know about the age at which the plaque is formed in general in the population. So I think what's happening is that it's, the soluble amyloid beta, it's not a problem. It's not a problem until the levels get too high, and then it starts to aggregate and becomes a problem. So I think the dynamics are similar in young and old. It's just that the young are in an earlier stage of this. And so, if you have the genes, and you're young, you're going to just be reaching that point earlier.
I'm going to show this to my colleague who's an eminent neurologist, and I don't think he knows it.
I don't think most people in the Alzheimer's field know this. I've talked to a few colleagues, and they're like, well, maybe those were the patients. Like they don't want to believe these data. These data were healthy, non-genetic affected. But it makes sense from the Alzheimer, from the amyloid hypothesis. It's not surprising theoretically. It's just that people studying Alzheimer's disease are so geared towards thinking that CSF amyloid beta 42 is a good thing to have.
All right, thank you. Next we have targeting aging biology for Alzheimer's disease prevention and treatment.
Is it on? Yes. Okay. Well, I know I'm standing between everybody and lunch, so I'll try to go fairly quickly. But thank you very much for the organizers for having me. This is a really fun opportunity. So this is like the slide whenever we do talks about Alzheimer's disease, we always have to show it about how devastating it is, how bad it is, how everybody's getting it. But I like to be a little bit more optimistic and really kind of cherish or think about all the things that the field has really achieved in the last few years. And so, one of the things that, of course, is really exciting is the emergence of these blood-based biomarkers. Now we're at the point where we offer precivity in the clinic. It's not covered by insurance, but I think that's going to be coming. And so I think, that is something that's really exciting. Of course, it correlates more with amyloid status, and this work has been done in symptomatic individuals, but still very exciting and more to come. Lots of research still ongoing in that field. And then other is our disease-modifying medications for better or worse. So our lecanemab, donanemab. And these drugs are so interesting because they're so good at removing amyloid from the brain. And they are maybe modestly effective at slowing cognitive decline. And I mean, we're glad we see some type of signal because we have to get people excited about our field and willing to go into it and invest. But I think it's really caused the field to kind of sit back and say, we were all told, we were all fed that it's amyloid that's, the driver of disease progression. So if we're clearing it and we're clearing it effective, and we're also seeing downstream effects on tau, why is it that we're not seeing disease or the degree of cognitive slowing that we'd like to see? And so, of course, one is that we're starting the treatment too late because A-beta accumulation happens so much earlier than other processes. And it will be super exciting to see the readout from the AHEAD study, which I know USC is participating in. So this is looking at adults who are cognitively asymptomatic but have elevated amyloid beta in the brain. So it will be very interesting to see whether that's more effective. The other, of course, is that Alzheimer's disease has lots of downstream manifestations, and amyloid is just one piece of the pie there. And we know that there are changes in tau and inflammation and microvascular injury. So perhaps if we're only targeting, this one small piece of the pie, maybe this is as good as we can expect for cognitive slowing. And then thirdly, Alzheimer's disease is just one type of pathology in the brain. And brains with pathology tend to be vulnerable to pathology overall. And most individuals who are older and have dementia have multiple pathologies. If we're just knocking out one, maybe we'll move the marker, the ticker a little, but not fully to the degree that we'd like to see in society. And so this kind of is a long-winded way to kind of go into the talk of rather than targeting specific pathology. I do want to be clear. I do think that we need to have biomarkers and know what we're treating. But whether we really want to be looking at individual proteinopathies, or do we want to be looking at things that are more germane to cellular or molecular functioning? And of course, we all know age is the biggest risk factor for Alzheimer's disease, no matter what else you kind of throw in that model. And that's the same for lots of other diseases too. Of course, everybody in this room is very familiar with these hallmarks of aging, another obligatory slide when you're at an aging meeting. And our work, and certainly many others, have shown that many of these same processes are implicated in accelerated brain aging and become more dysregulated in neurodegenerative disease. And the reason why this may be important is because, at least in animal models, of course, there have been a lot of interventions trying to specifically target these different pathways, and some of them have been shown to be effective in increasing lifespan and health span, although, of course, we do need more data from humans. So cellular senescence, there's been a lot of interest and talk around cellular senescence and the role that it might play in pathology really across the body, since we know that the burden of senescent cells goes up across tissues as one ages. And I know Jim Kirkland is coming a little bit later today and will be speaking, but his work was very instrumental in the field in terms of really using a hypothesis-driven approach to find medications that selectively target senescent cells, so working on the anti-apoptotic pathway. And so there's been many of these compounds that have been identified, and there are newer ones that are coming out, but at least to date, kind of the most well-tested ones are the combination of dasatinib and quercetin. So dasatinib is a treatment for certain forms of leukemia that's FDA approved, and then quercetin is a flavonoid available over the counter. This is work by my colleague Miranda Orr, who I imagine many of you know. So here she is doing single-cell transcriptomics within the hippocampus from individuals who have passed away. And what she's showing here, that it's specifically the neurons with the neurofibrillary tangles that seem to show upregulation in genes that are associated with senescence, so suggesting that there is at least some type of association between neurofibrillary tangle accumulation and cellular senescence. And this was shown more eloquently in experimental trials, although, of course, this is mice and not humans. So these are pretty old mice. They were 20 months of age. They had wild type and tau transgenic mice. At this age, these mice have really diffuse neurofibrillary tangles, and they were treated biweekly with dasatinib and quercetin or with vehicle. And they found specifically within the tau transgenic mice that there was about a 35% reduction in neurofibrillary tangle burden with senolytic treatment. And There's been a lot of other studies looking at senolytics for lots of different conditions, so we have safety data on it. So when Miranda and I were young and naive and early professors, we decided, why don't we do a clinical trial? How hard could that be? And it turns out it's very hard and it's very expensive. And we only had $50,000 as a pilot study. So we enrolled 5 participants. Demographers, don't freak out here. I understand it's very preliminary. And so this is an open label study. And we were very mindful of the fact that, the effect sizes are going to be, small and variable. And so our primary aim was really to see whether we have evidence that these medications are in fact reaching the central nervous system. There's some data from oncology, but not enough in people that don't have brain cancers. And then we wanted to also get additional data on target engagement and safety and cognitive and physical function markers as well in order to be able to better power future studies. So this was published last year. And again, small sample size, open label. So we are moving forward with a phase 2, but ultimately we found that the treatment, it was safe, it was well tolerated. There was evidence that dasatinib at least penetrated the central nervous system. We did not see the same for quercetin, so we're not sure if that's clearance related, that we just weren't getting that time window right to be able to detect it. More work there needs to be done. We saw some evidence of lower senescence markers in plasma and CSF, and we didn't see any significant changes in our biomarkers, and probably not surprisingly, we only had 5 people. But at least in CSF, using the Lumina pulse assay, I will say, we saw at least a trend towards increasing A-beta 42. So potentially a signal there, but really the proof is going to come down to randomized control studies. Okay, so next drug I'm going to talk about is metformin. So metformin, as everybody knows, is a drug that there's so much interest in the aging field, and that's because it has so many downstream effects on lots of these like canonical hallmarks of aging. So things like cellular senescence, and of course its impact on energy, metabolism, and reactive oxygen species. And there's been a lot of data, and I will say conflicting evidence too, about whether or not metformin may or may not modulate Alzheimer's risk, and whether it does in the directionality that's desired or not. You see a lot of heterogeneity in research studies, but there is thought, of course. We know that midlife type 2 diabetes is a risk factor for developing Alzheimer's disease. There's at least data to suggest that metformin can lower inflammation, so perhaps through its role on reducing reactive oxygen species or maybe leading to better integrity of the blood-brain barrier, perhaps that could be protected. So there have been some kind of small trials, and there is a larger, much bigger study called MAP that's going on, which is like a nationwide study trying to see if metformin treatment is able to slow progression of cognitive decline in people who are symptomatic. Okay, having done one clinical trial, or actually 2, I learned from my mistakes and I realized how hard they were to do. And so we got a little smarter with this next grant. So this is a grant that I have with Dr. Sarah Espinoza, who will be here later, and Dr. Nick Mitzi. And so this is a grant where we're actually using entirely data that's already been collected. So Sarah Espinoza and Nick Mitzi, they had a previous grant where they were comparing metformin versus placebo for frailty prevention. So enrolling older adults who were pre-frail, and they were treated for 24 months. And then in addition, the other nice thing is they got a second grant, and they were able to assay lots of different biological aging pathways. So this really seemed like a prime opportunity to be able to test some of our hypotheses in a way that is at least conscientious in regards to time, money, staff, and participant burden. So what we're doing is we are running new assays on this data. We're looking at lots of biomarkers in blood. We're seeing how they associate with biological aging hallmarks at baseline. And then we're going to see if metformin does actually modulate the trajectories of these biological aging pathways. Does this do anything to change in ADRD biomarkers over time? So this grant was recently funded, so I don't have any data yet, but hopefully in a few years we'll have some more results. And then so far I've talked about drug trials, and I think certainly drugs have to be part of the solution to combating Alzheimer's disease. But now that mindset is shifting and we're thinking about Alzheimer's disease as really something much more of a life course disease. We may not want to be on these drugs forever. And so also kind of some interest in whether if you can actually kind of mimic some of the beneficial effects by doing things that are more like exercise or focused on nutrition. And I think a lot of this work came out of USC showing that kind of periodic fasting kind of doesn't really matter what kind of species you're looking at, all the way down to yeast, human, you tend to see increases in lifespan. We know those people that live a really long time in the blue zones, they also tend to be people who consume less calories overall. And so there's been a lot of interest in just whether periodic fasting or intermittent fasting, if it might be able to extend lifespan and also reduce burden of chronic illness. And there's also been a lot of interest in animal models trying to assess this in association with neurodegenerative markers, suggesting that maybe this intermittent fasting, it's going to reduce inflammation and improve metabolism. Could that have beneficial effects? Could it be tapping into the mTOR pathway and also lead to changes in autophagy? Or could we also have changes in the gut microbiome, and that could also have associations with better cognitive outcomes and prevention or treatment of ADRD. So we have another study that we fondly call NIBBLE. It's our nutrition intervention for body, brain, and longevity effects. And so this is going to be a 6-month intervention trial with the fasting mimicking diet. And we're going to be recruiting individuals who are middle-aged, ApoE4 positive, and we're randomizing them to normal diet versus the FMD. Our primary outcome of interest is to look at changes in cerebral blood flow. And these are always the tricky thing with these studies where you're trying to go earlier, is you don't have so many abnormal markers to change. So this study, it's on clinicaltrials.gov. We're recruiting, but of course, no data to share yet. And then just to kind of give you a whirlwind overview, we're also launched a new study too, which is an observational study. And we're really interested in looking at these different hallmarks of aging. I think we're fortunate to have a good team in Geroscience and a good team in ADRD, and we work closely together as a team. And so we're really trying to understand this much more systematically and also to validate some new biomarkers. But we're just starting, so we have a very long way to go. And with that, I just wanted, it takes a village to do all of these studies. And so big thanks to all my colleagues at Cedars-Sinai, UT Health San Antonio, where some of this work was done, Miranda Orr, who's been a very important colleague, and then of course, all of the funding institutes. So thank you for your attention.
So thank you for the talk. I think it's very exciting to use these intervention studies to try, to slow the progression of dementia, but also to understand whether these have anti-aging effects in general. So I have a question about how we think about the biomarker measurements. When you mentioned epigenetics as one of the biomarkers that you would measure, say in your metformin trial. All these aging biomarkers tend to be like surrogate measures, composite measures of multiple biomarkers. So how do we think about effects on individual biomarkers versus the effect on composite measures?
Yeah, I think that's a really good and that's an important question. And it's so hard for us to test because The only way you can look at these individual markers is if you have a ton of data, right? Like, because otherwise you're doing lots and lots of comparisons, and you're probably, once you correct for all of those, if you have like a randomized control study, you don't have a whole, 20,000 people in your study. And so I think that's a real potential limitation of the clinical trial approach. And I think that's why we need to work with our friends in population health and try to understand, what signals are being seen in these, really large data sets where you can control, and then actually go back and empirically test it in the clinic. So I definitely think more work needs to be done in that regard. So I completely understand and definitely agree with you that that's the way a field needs to go. And I also think that we can't just treat everybody with everything. Like, we do need to stratify people based on having specific biomarkers that are abnormal. That answer your question?